back. Laser pointer. Yeah.
Okay, everybody, welcome back to the first session of this afternoon. We have in our panel Dr. Olivier, as you know, from the University of Brest, Dr. Lohans from Sorbonne University, and Dr. Per Albert from Gothenburg University. Okay, so as you probably already know, you have 15 minutes to present, okay, and then 15 minutes for discussion, okay. So, Carmen, you have the floor. Thank you very much.
Good afternoon, everyone. My name is Carla Lopez, and I'll be presenting my master thesis, which is titled The Suitability Assessment of Alginate Immobilized Microalgae for Aquaculture Wastewater Treatment and Biomass Valorization for Biodiesel. I'm going to start by briefly introducing the topic, which is centered in aquaculture. Aquaculture is one of the fastest growing food production sectors nowadays, and it currently provides around 60% of all the seafood for human consumption. And it's expected to increase by a further 60% in the next 30 years. But with more fish farming comes more residues produced. In aquaculture wastewater, the nutrients or the contaminants present here are in significant amounts of carbon and nutrients, which are in the form of organic matter, ammonium, nitrates, and phosphates. Considering that most of the effluents are discarded or discharged into natural streams, if they are left untreated, this can cause eutrophication and anoxia, disrupting the ecological balance of aquatic ecosystems. Therefore, the use of microalgae in wastewater treatment comes as a natural response because they can enable the recycle of nitrogen and phosphorus, but while also generating biomass for biotechnological applications. These are two of the most popular species utilized for this, so Nanacrodopsis species and Tetraselmi species, which are preferred because of their rapid growth and environmental resilience. These are the reported nutrient removal efficiencies for both species. Nevertheless, most approaches are based on free cultures, which have a main problem because of the harvesting of the biomass. Therefore, immobilization comes as a possible solution for this. Some of the advantages of this technique are that now the algae is shielded in a bit, therefore preventing from chemical and biological hazards, while allowing continuous tertiary treatment. Some of the disadvantages are that this, of course, includes a manufacturing cost, extra cost, and the limited lifespan of the beads. Therefore, if we can integrate aquaculture and microalgae, we can have circular resource, bioresource production, because the biomass can be used for feed, nutraceuticals, and biofuels. Therefore, the main question of this project was, what is the potential of immobilized microalgae to remove nutrients in aquaculture wastewater and accumulate high-quality biomass? The main objective was to evaluate then two immobilized microalgal strains, Nanacrodopsis oceanica, CCMP 1779, and Tetraselmis convolute, BS14, for nitrogen and phosphorus removal and biomass production. The hypotheses here are that immobilized cells are going to exhibit a positive growth while also high nutrient removal. This nutrient removal is going to be driven by strain-specific N2P uptake ratios, and environmental factors determine the lipid profile, the final lipid profile. To assess this in more detail, we immobilized algal cells in alginate matrices. We evaluated them for nutrient removal and growth under two photo periods. First, 12 to 12, and then 24 to 0, light-dark regime. First under a closed batch system, and then in a continuous flow system. Then we quantified the biomass production in the continuous flow system and also analyzed the fatty acid methyl ester profile for biodiesel applications. So for the methods, we first started with the cultures of the two strains for 7 days to establish the growth curve. Then we scale up this in semi-continuous culturing to keep them in exponential phase. Then we centrifuge these to obtain a concentrated microalgal suspension that was then mixed with sodium alginate and sodium chloride. This mix was added into a burette so that it can be also added dropwise into a solution of calcium chloride. Then they were left here for an hour to stabilize and form the beads, and then the beads were washed several times. Finally, we ended up with our bead stock that also included, of course, the two strains and the blank beads as a control. For the closed batch system, we established two trials. One regarding the 12 to 12 photoperiod that included one blank, essentially with only 1 liter of wastewater, then another control with only blank beads, and then the treatment controls, the algal treatment controls, which included 100 grams of beads plus 1 liter of aquaculture wastewater. These were cultured for 4 days. Air bubbling was applied at 18 degrees and a light intensity of 152 micromole photons per meter squared per second. The setup was the same for the other experiment, except that this time we included 200 grams of beads and 1 liter of aquaculture wastewater. The environmental parameters were the same, and the light intensity this time was a bit higher. The aquaculture wastewater was sourced from a local fish farm. The analytical methods included the sampling of the water and the beads every 24 hours. The water was filtered for nutrient concentration analysis, and we used this equation to analyze nutrient removal efficiency. Then the beads were dissolved to assess cell density using a Neubauer chamber. Cells per beads were assessed using this equation, and growth rate with this other equation. Now, our results for nutrient removal under the 12-hour photoperiod in a batch system showed a gradual removal for ammonium, nitrite, and nitrate, but the results were not conclusive for phosphate removal. Letters here indicate statistical differences, and as you can observe, only for ammonium the algal treatments managed to outperform the blanks, which we were expecting, but for the removal of nitrite and nitrate, there was no difference between the blank beads and the algal treatments. And this can be explained because we didn't sterilize this water, so of course there's going to be microbial communities here that can be causing nitrification, so therefore reducing the nutrients of nitrogen. And for the phosphorus results, we can observe that the blanks actually are more efficient in removing the phosphates than the algal treatments. But we thought that this might be explained because of phosphorus precipitation, which can occur in the presence of magnesium, calcium, and iron ions, which are also high in seawaters. These are the results for the 24-hour photoperiod that are similar, but now we can observe a steeper removal of nitrogen species occurring after only 24 hours. And we can observe an improved removal of phosphates. The strong activity of the blank beads is only observed in ammonium, and now all of the algal treatments outperformed the blanks significantly. Now we were interested in explaining why are we observing this lack of removal of phosphate, or limited, or not as much as reported in literature. And we can explain this by the N2P ratio, by the NP ratio in our system, which here is 9.3 to 1. That is already suboptimal considering Redfield ratio as the optimal. Therefore, our system is already depleted in nitrogen, but also this is a batch system. So considering that there's no new inflow of water, this might be causing nitrogen starvation in our system, which directs the cells to produce lipid and starch rather than DNA and proteins. These are the results for growth rate under both photoperiods in the batch system, and we can see that the cells are growing exponentially in both strains and in both conditions. Nevertheless, the growth rates are presented here per day, and these are lower than reported in literature. Additionally, we can observe that under the 24-hour photoperiod, the growth rates are actually a bit lower than in the 12-hour photoperiod. Then, why are we observing the lower growth rates for these results? This might be caused because of the high encapsulation densities. So probably our centrifugation or the initial state of the cells was too strong, then causing some sort of self-shading inside the bead. Also, we didn't add vitamin addition to the cultivation in this aquaculture wastewater, which we know microalgae might be benefit from this. Also, the lower growth rates under the 24-hour photoperiod can be explained due to oxidative stress, due to continuous elimination. Based on these results and the fact that the Tetraselmis convoluted ES14 species showed no difference or like little decrease under both photoperiods, and considering the fact that this strain is local, we keep this as the species to use in further experiments that included the setup of a continuous flow photobioreactor. The photobioreactor was connected to an influent tank and an effluent tank to allow an exchange of water of 1 liter every 3 days. The photobioreactor included the beads entrapped in a cage to prevent them from escaping. Also, light intensity and hemoglobin was applied. And 3 trials were run. The first one for 4 days under a 12-hour photo period, the second one for 4 days under a 24-hour photo period, and the last one for 9 days under a 24-hour photo period as well. The analytical methods were the same, except this time the water was, the filtered water was also used to analyze total organic carbon. The cells, the dissolved beads were used to analyze optical density, and at the end of each trial, all of the biomass was collected, dissolved in sodium citrate, to quantify volatile suspended solids and FAME quantification of lipids. This is the graph for the results of neutron removal under the continuous flow system for the both photo periods. And as you can observe, there's also high removal efficiencies for all of the nitrogen species. Particularly now for the 12-hour photo period, the system managed to remove almost all of the nitrogen concentrations after just 48 hours. And for the 24-hour photo period, this occurred similarly as in the batch experiments, again under only 24 hours. The removal of phosphates are positive for under the 24-hour photo period, but not precisely amazing for the 12-hour photo period. Now, how can we explain the efficiency of the systems or the improved removal efficiencies of the system? I think it's because now there's a continuous flow system, so the process of nitrogen starvation is not being observed here. Thus, also the NTP ratio is partially stabilized. And it's also important to notice that these experiments were carried at room temperatures, which in spring can be quite high in Naples. And yeah, so more temperature means more metabolic activity as well. These are the results for growth in the continuous flow system for both photo periods. And we can observe a linear increase for growth and for biomass. Particularly under the 24-hour photo period, the growth is higher. But it's also important to notice that on day 3, the growth and the biomass start to decrease. Comparing these results to the literature, these results contain actually a higher biomass productivity than previously reported. We can explain the positive results because now we're using a more stable system, a continuous system, that is actually more realistic. And also the biomass production that we found here was also seen in other reports, but these studies were using actually free cultures. So this might offer an operational advantage of the beads. Finally, the fatty acid methyl ester profile was not conclusive for biodiesel production because it has a higher concentration of palmitoleic acid and linoleic acid, which are polyunsaturated fatty acids, and a lower level of saturated lipids, which are palmitic acid and stearic acid. This means typically for biodiesel production, we prefer saturated lipids, but we didn't find this profile here. This means that the high profile in polyunsaturated fatty acids might be more suitable, therefore, for aquaculture feed or nutraceuticals rather than for biodiesel production. Finally, the conclusions of this project are that aquaculture wastewater bioremediation was demonstrated by immobilized Tetracernis convolute and Anachroropsis oceanica, particularly under continuous inflow and illumination. There was also high biomass yields despite signs of oxidative stress after day 3, as I mentioned. And there's a potential application for feed and nutraceutical value rather than for biodiesel. As some of the perspectives for this project, we might want to consider trade-offs between choosing one or other photo period, particularly because we know that under continuous illumination our cells are experiencing oxidative stress, but our results show that this also accumulates the high biomass. So we might want to choose between a more stable and gradual photo period and growth, rather than probably a more stressful for our algae. Of course, to maximize biomass and the lipid production. Another perspective would be to test different hydraulic retention times, because we stick only to the 3 days, which means 1 liter of water exchange every 3 days. So we might want to focus on reducing this HRT to see if our system managed to remove the same amount of water in less days. With this, I thank you all so much for your attention, and all questions are welcome.
Thanks very much, Carla. I'll give the floor. Do you want to start, Carla?
Yeah, I can start. Thank you very much for this presentation. How is this going to be able to scale up? How is it going to work? I didn't understand how this little experiment can be done with a bigger.
So, well, this is one of the aims of this study is, of course, to evaluate its applicability.
Yeah.
And I think the main strength of this relates on the photobioreactor itself. Because, well, nowadays, wastewater treatment systems and aquaculture wastewater systems as well include currently bioreactors. So this would be an addition, and additionally, this could be an additional photobioreactor, including the entrapped beads. Therefore, we can also maybe explore to putting them inside a cage, but like essentially the system is following the flow of water that a natural aquaculture system would have.
Yeah. But this aquaculture system, you said something about open system. How do you? Is that when you have them in the sea directly? And how do you get the wastewater down? I didn't understand that.
Okay. No. I think this is based in inside aquaculture systems. So this is already indoors, and you have like different tanks, and of course you have like the pumps that bring water from the sea, essentially. So the water is being it's passing through the fish tanks first, and then there are certain tanks already, including bacteria and other techniques. So like this photobioreactor could be inserted into one of these processes.
And this technique with the beads, you said something about that they need to be replaced, or
Yes.
So what's the lifespan?
Well, yeah. Particularly for these experiments, so we did the batch experiments first, if you remember. So these ones.
Yeah.
And after this treatment, the beads were after the last trial for the closed batch system, the beads were stored actually, and these ones were the same beads that we use in the continuous flow system then. So I think we can observe a decline. So the lifespan of these beads might be so after the first two trials, so after 8 days, and then they underwent a light period, and until this day, so day 3 of the continuous experiments, they started to decline. And well, particularly I have this in some annexes, but this is the run over the course of 9 days, and this is day 3. So we can also observe that the beads decay drastically after this. So we might consider this, lifespan.
Yes, thank you, Carla, for your nice talk with, I'd say, an Italian rhythm to the way you talk.
Sorry.
I continue on what you were saying. The length, or let's say the biomass with the other algae was similar to the one you picked up, or it was different? It was from your previous the trial, the initial trial.
Yes.
It looks like if for the other one,
Nanochlorosis.
Nanochloropsis. Did you have more cell densities, or
Yes.
and they remained for longer time, or not?
So the growth rates, I actually tested this with the statistical test, and the differences between the algal treatments and the photoperiods are not significant. So this means that both microalgae are like growing consistently
Yes.
and similarly. And these differences, like this is higher because we started from different initial cell densities.
Okay.
So,
And what is the reason for starting with different cell density? It was because the culture were like that?
exactly.
Okay.
Basically.
Okay. So now I understand why there are those different
Yeah.
Okay. So moving to the applicability, I think it's really nice to see that first there is a proven efficiency, and the way you have shown how you calculate the nitrate and ammonium removal, etc. So that's very well evidenced. For the phosphates, even in traditional wastewater plant, there is an issue with phosphate.
Yeah.
So, can you tell us what do you see as advantage to remediate phosphate with algae instead of with microorganisms? Yes, with all the also CDS etc. for phosphate. What do you see as an advantage of using microalgae? So the beds.
Yeah. Well, for the removal of phosphates, we found limited and consistent results. But I do think that one of the scopes of this project is to apply this biomass to the valorization of other byproducts. So particularly microalgae is reported to have like high contents of lipids that can be then applied to other biotechnological.
And so, in terms of lipids in the microalgae,
Yeah.
in the literature, you told us a little bit.
Yeah.
Can you tell us what are the conditions that maximize the potential for biodiesel production or for total lipid amount or flames concentration
Yeah.
in phytoplankton? Do you have an idea?
Yes, I actually mentioned it briefly with the nitrogen starvation. So it's been reported that actually if algae is grown in limited amounts of nitrogen, this maximizes the lipid extraction.
Yes.
So I think the main strength in this project relies on using natural aquaculture wastewater. We did nothing to this, so and it managed to show good removal efficiencies. So this came just as a possible valorization for this biomass. But of course, if we would want to synthesize lipids in a scaled up process, we would include nitrogen starvation for a pre-treatment of this aquaculture wastewater.
Yeah, that's for sure. Normally, the maximum of production is more in the exponential phase than in the stationary phase than in the exponential. And I wanted to ask you why you presented the famous results as percentage only and not quantity related to the biomass or something like that.
Can you elaborate?
Like your on the results on the phase, it's we have the percentage of the different compounds.
Yes.
And
No, the lipids.
no, the famous results.
Okay, yeah.
Yes, you see, this is the percentage of total FAMES.
Yes.
But in fact, in terms of potential, it is the quantity that is more relevant to.
Yes.
So maybe you can tell us what is the quantity that you measure in those two trials.
Yes, it's here in a very
Okay, I have not seen it.
It's the total
Okay.
content per dry per cell weight.
Ah.
Yeah, sorry,
Ah.
I didn't discuss this.
Okay, and this is yes, that's better. Okay, cool. And about the potential for using the beds, because the problem is when you upscale that, you have to, all those beds, you have to treat them. Maybe they won't be, or it's exponential phase, so maybe for lipids that not be the best way. But you have the alginates and then all the polysaccharides of the algae. Can you think of another biofuel that you can produce with those nitrates?
Well, I think I'm not very familiar to that topic, but I know that there's been some efforts in including anaerobic reactors for the production of, I think, methane and other gases as source of energy.
Or bioethanol with polysaccharides? Very interesting. Olivier?
Thank you. Thank you, Carla, for your presentation. As you know, this is far from my area of expertise, but I did enjoy your presentation. It was well constructed. Really, your slides are very good to illustrate that. But as Hans mentioned, I like the Italian tempo. Maybe you had a minute left at the end, and you slowed down. You started very fast, and you slowed down a little bit as we were going over the. And the thing is, you had time to explain some things a bit slower, like the axes of the graphs, the colors, the total thing. A few things like that escaped us because we're trying to listen to the very interesting stuff you're saying, and at the same time we're trying to understand the figures. So that did not help me also. Although I'm used to evaluating and seeing presentations, as it's far from my subject matter, it was a bit fast. So that would have helped me. But nonetheless, you managed to get your message across. So it could be even better. So work on that in the future.
Thank you.
I do have one or two probably a bit naive questions. In the closed batch system, you have two photoperiods.
Yeah.
But not all other variables or experimental parameters are kept constant. Why? Like for example, the light intensity is not the same.
Yes, that was a constraint of the material that we had access to.
Okay, it's not a design decision. It's just because you had no choice to do that.
Yeah, exactly.
Okay. Because you realize it does make your interpretation
A bit
more
hard.
difficult. In your case, it's pretty well cut, so not. But that's something we try to avoid.
Yeah.
And were other variables other than the light intensity different in the two photoperiods?
For the batch system?
Yeah. No, it was you. Yeah, that was your
only
one.
the light
Okay.
intensity.
In the intro, where you rented the context, you go over the pros and cons of, advantages and disadvantages of. You say, well, the lifespan beads have a limited lifespan, and I guess compared to free cultures. Why?
Yeah, this is actually assessed by the total organic carbon content. So I did not show those results, but we have it here. So the total organic carbon is an indication of the alginate degradation, actually.
Okay.
So we can see that, that over the course of the experiment, the total organic carbon increased exponentially. So for both photoperiods. This means that naturally the beads are gonna dissolve.
Yeah, but compared you say they have a shorter lifespan compared to free cultures. What would happen in the free culture? That's what I mean. And also, what is the approximate reason why they have a shorter lifespan? Now you're showing they have a short lifespan. Okay, we can agree on that. But why is it like that? Do you have any idea? I know it's not the questions you were asking, but
you mean the beads, like the
why do they have shorter lifespan in bees than as a free living organism?
Well, with that statement, I meant the lifespan of the beads, not the algae itself.
Okay, it's the lifespan of the bee.
Yes.
Okay, and it's a very applied project. You demonstrate that a lot of things can be done and can be done better in terms of extracting and assimilating, etc. Was the question of the basically the cost benefit trade off in terms of this has economic application directly? Was this something you approached at all in the thesis or not at all? And if not approached directly, do you have any idea? Do you think it makes sense for a business to do it compared to what they're doing with free living cultures or not?
Well, I think it has its pros and cons. For example, for the free cultures, I think there are some costs included in the maintenance of the photobioreactors, for example, which also can be applied to this one. But I think particularly the free cultures offer a limitation in an aquaculture system because of the harvesting. So I think this could be an advantage for applicability. So the cells are, so you could be building more beads and then harvesting the beads, particularly with these experiments after even just 3 days. So that's like, now you have also the trapped biomass that can be directly assessed in the lab. So, but I think, of course, there's like, so I know, for example, that for free cultures, you have to, in some systems, you have to stop a little bit the system and then harvest that biomass, and then, of course, centrifuge a huge amount of culture. So with this, this is like continuous treatment, so you could not stop the system and then just harvesting beads.
If the team wants to patent and sell this, we need to evaluate the cost benefits in terms of.
I will think about it.
Think about that. All right, thank you very much.
Thank you. It's PDF, yes.
Okay, everybody, so now we're gonna talk about puffins. Emma, so the floor is yours, good luck.
So hi, I'm Emma, and I'm really excited to talk to you about my master's thesis today. It's about Eurasian otters in Atlantic puffin colonies, and I'm looking at the temporal and spatial patterns of activity and predation on 3 different Norwegian islands. So seabird populations are declining globally due to multiple interacting pressures. The main ones that are often cited as contributing to their population declines include climate change, extreme weather, pollution, overfishing, and predation. When it comes to predation, there are a lot of different predators at play that either prey on the seabirds themselves or their eggs. And we know a lot more about invasive predators like the American mink or rats or domestic cats than we do about seabirds' native predators. So I'm interested in looking at a native predator in Norway, which is the Eurasian otter. And these otters are expanding their territory after near extinction a few decades ago. They're also considered fish specialists, but they've been documented to eat other things like invertebrates, small mammals like rabbits, and other species of birds. The prey that I'm particularly interested in are these 3 species of auks. This includes the Atlantic puffin, the common guillemot, and the razorbill. These 3 birds nest in slightly different ways. The puffin lays an egg inside a burrow that it digs out of the cliffside, and the other 2 species lay their eggs in rocky crevices and ledges. And another big difference is that the puffin chicks remain on land for a lot longer. They remain on land for 6 to 8 weeks compared to 2 to 3 for the other 2 species. So I'm mainly focusing on the puffin as a prey item, or potential prey item. And puffins spend most of their year at sea, and they only return to land in the spring and summer to breed. They're also endangered in Europe. So I have 3 main objectives in my study. The first of that is the temporal activity. I'm essentially asking the question, does otter detection frequency vary across puffin breeding stages? And my second objective is about spatial activity, and I'm asking whether patterns of otter activity vary spatially within the puffin colonies and also between the islands. My third objective is pretty simple. I'm basically just trying to confirm whether these otters are eating seabirds, and in particular puffins, and if they are, when they're doing it, where they're doing it, and how often. This is my study area in Norway. I looked at 3 different islands. Jamsoya and Hornøya are in the north, and Runda is closer to the south. And we set up between 17 and 19 camera traps on each of these islands in the puffin colonies. And these cameras were running for over a year in all cases. We were able to start them at a different time in the 2024 breeding season. But we captured one entire winter and one entire puffin breeding season in 2025. This is what the study design looks like. The puffins are breeding all over this cliffside, including in the brown areas on the left side of the graph. But realistically, we could only monitor a subsection of the colony. So that's what's shown in green. And the blue line is a possible access route into this colony area that an otter could use. And we placed cameras at all of the possible entry and exit points of this sub-area of the colony. And then on the right here is to just illustrate the sort of steepness of this cliffside. And that's the reason that we don't have any cameras on one side of this colony area. It's because it's essentially inaccessible due to the steep cliff. And we had 3 colony areas like this per island. So once our study period was over, we collected the data from our total of 55 cameras. This generated over 500,000 images, which I then put into a software called Agouti, which is really great because it separates all of these images into sequences, and each sequence contains all the images of whatever animal tripped the camera to go off. And then I could go and click through each image in each sequence, and I could annotate what species was there and what behavior it was doing as well. So I wanted to walk through what I expected to see in my data after I had all of this back. This is a graph containing fake data just to illustrate my hypothesis, which is that the frequency of otter detections will be higher during the puffin breeding season. And in this graph, we have the date on the x-axis and detection frequency on the y-axis. The detection frequency is of the otters, and it represents the number of detections of otters per camera per day in each of these colony areas. And the shaded blue indicates different stages for puffins breeding. The lightest being when no birds are present on land, they should all be at sea. The next one being the non-laying period, when adults are back in the colony, but they haven't laid any eggs yet. And then we have incubating and rearing. And basically, I'm expecting that the detections will peak sometime between incubating and rearing. My idea here is just that during this period, there are actually more adults in the colony, and there's also eggs, and there's also chicks, so there's a lot of potential food at this time for the otters. And I also expect that the otters won't be in these areas as often during the time when there's no birds there. My next hypothesis is pretty similar. Again, this is just fake data to illustrate my idea, but I'm also expecting that if the otters are predating these birds and I'm going to catch them on camera, I'm going to have more instances of that during the incubating and rearing stages, rather than the non-laying period, which I might see some. There's still birds there, and I don't expect to see any when there's no birds. And I just defined a predation event as a sequence of images in which an otter is carrying or actively eating a seabird. So for my results on temporal activity, otter detection frequency did vary significantly with breeding stage, which I determined through generalized linear mixed effects models. And the strongest differences in temporal activity occurred on Yumsoya. These are my detection frequency graphs for Yumsoya. They have the exact same structure as the one of my hypothesis, but there's one for each colony area. And as you can see, what happened was not what we expected. There was actually a significant decline in detections during the incubating and rearing stages for the otters. And there was a significant increase in detection frequency during the non-laying period when the adults had just returned back to the colony. And then interestingly, there was also a really strong peak in the guillemot field, one particular colony area. And this was really interesting because it also coincided with images of the seabirds coming back to land. And there were also some images of otters predating the birds at this time. And this is new, actually. The people who were working in this area didn't realize that the birds were coming back this early. That's also why it's occurring during the time when I said there shouldn't be any birds there. But yeah, this is just an interesting part of the data. My next objective was spatial activity and otter detection frequency also varied significantly between colony areas within islands, and it also varied between islands. So Yamsoya, which I just showed you, and Hornoya, which I'll show you next, were more similar in their patterns and frequencies compared to Runda. These are the graphs for Hornoya, and what I want to illustrate here is that the detection frequency was much higher in certain colony areas, such as Puffin Field 2. They were there like very consistently throughout much of the year. And then in other areas of the colony, they didn't really seem to use it as much. This is Runda. Unfortunately, we had very low detection frequencies in Runda. We only had, I believe it was 12 detections throughout the entire study period, so there weren't many patterns to go off for this. My final objective, which was predation, I can confirm that the otters are predating auks, including puffins and guillemots, and we had 11 instances where they had caught adult birds and only 2 instances where they had caught chicks. These chicks were later in the season when they were larger. And most of the predation events that were recorded were during the non-laying period when the adults had just gotten back to land, and that early season peak that I just showed you on Yamsoya. And then again, there was a lot less predation witnessed on our camera traps during the time of year when we thought would be the highest, which was the incubation and rearing period. Next, I just wanted to walk through sort of an idea I have to explain the temporal activity specifically in my data. And the idea is that seasonal changes in marine prey populations might be driving otter activity and predation in the puffin colonies. So essentially, in the spring, we have a situation that looks like this. There are adult puffins that have just come back to land to breed, and they're up in the cliffside, but there's also studies showing that the marine prey, the fish in these areas might be at a lower abundance compared to other seasons. And some studies show that Eurasian otters will actually switch to less profitable prey that needs more handling time or has less caloric benefit to them compared to what they normally eat for fish during the springtime. So the idea is that the otters that are next to the seabird cliffs might be taking advantage of the seabird colonies during this time that might be sort of a stressful period for them. And they're deciding to go up into the cliffs more often to hunt the puffins rather than go in the sea to catch fish. And then conversely, we have a situation in the summer where there are more birds back on land breeding. There's eggs available and there's chicks available, so there's a lot of potential food here for the otters. But there's studies to show that there also might be more fish available at this time in the summer. And so the idea is that the otters are spending more of their time catching the fish that they prefer instead of going up into the colony as often to hunt for the puffins. My next point of discussion is whether these individuals are exhibiting diet specialization. And so some Eurasian otters have been shown to have individual prey preferences, even between otters that are foraging in the exact same areas. Individuals may consistently choose specific types of fish. And there's also anecdotal evidence of otters specializing in things like crayfish, crabs, and in one area of southern Norway, eider ducks. But for my study areas, more research is definitely needed to determine whether individuals are becoming specialized on puffins, because with my camera traps, we can't distinguish between individuals. All the otters essentially look exactly the same. Sometimes we can say whether it's a male, sometimes we can say whether it's a female if it has cubs, but it's pretty hard to determine who's who. So yeah, more research is needed for that. My conclusions are that otter activity was colony specific, and this means that the patterns that we might see in one seabird colony or on one island aren't necessarily something that we can extrapolate towards another colony or area. And the period of vulnerability for puffins was also earlier than expected, especially on Yamsoya. The puffins that are returning to land are the most vulnerable to otter predation, it seems like, compared to in the summer when they're incubating or rearing. And also, most of the predation events involve adults, and this is a potentially important result because these seabirds are long-lived species and they have a very low reproductive output. They only lay a single egg and they only have one chance to breed. So in populations like this, adult mortality can potentially have a disproportionate effect on the population as a whole compared to the occasional loss of eggs or chicks. My final conclusion is that year-round monitoring reveals patterns that might be missed by studies that only look at the breeding season. Many studies on these seabirds in general, but also on predation, will only look at the breeding season, and they'll only look at the part of the breeding season where we expect the birds to be there as well. So when you have a year-round study, you can see more patterns that are maybe happening in the wintertime. And then my final conclusion is that Eurasian otters are not the main driver of puffin declines. We shouldn't be running out to these islands and putting up fences and trying to keep the otters out right now. But they might be adding a localized pressure on these seabird populations that are already struggling. And that's everything.
Thanks very much, Emma. Olivier, do you want to start?
Sure, why not? Thank you very much, Emma, for this nice presentation. I'm shocked that even as they are chewing on the skull of a just dying bird, they can still look so cute.
They are.
And looking directly at the camera.
It's
as well.
Yeah,
Wow.
they really do like to
So
check out the
photogenic,
cameras.
it's
Yeah.
incredible. I really like, one thing I really enjoy about your work is, and you said it, it shows the importance for answering some ecological question, the need for these continuous monitoring of population and colonies, because what the things you've shown, it could not have been shown any other way than with these kind of long-term study, and that's super interesting. Did you go to the field or did you do
Yeah, so these cameras were obviously set up before I
Yeah.
even started this master's. But last year I did get to go to the field in Yomsoya. This picture is actually, I took this there.
Okay.
I did get to go to the field there and help out with just some of the seabird research that they were doing, but I got to see all the locations of all the camera traps. And I just checked on all the SD cards and stuff. And then I got to go to Runda as a tourist, just to the viewing station. So I didn't see my camera traps on Runda because there was no one to show me around and I didn't have access there. But yeah, Pornoy is
You
the only
managed
one
to go
missing.
into the field and they
Yeah.
did show you around, that's nice. I'm wondering, in your, you had to look at the 38,000 sequences, if I understand. You annotated everything. Did you see any other interesting things
Yes.
that you did not mention? That,
Food or other
well, I was
predators?
wondering, other predators or other prey actually, exactly the two I'm wondering.
I can show you if you'd like.
Sure.
Let's go to the end of this. My secret fourth objective, other predators, which is in my thesis, but
Okay.
I had to cut. We found some American mink. This was an interesting sighting because these are actually trapped on this island. They're not supposed to be there. And then we found stoats, particularly on Hornoia. This one's rolling an egg away. These are domestic cats on Runda. It's a little hard to see, these ones are at night, but the one on the left has a puffin in its mouth, and this one is looking directly into one of the burrows. These are rats. There were a lot of rats on Runda. This was my coolest, the coolest part of going through this, even though I love the otters. There were a ton of white-tailed eagles, and they were also predating the birds a lot, considering this study was not, meant for avian predators. There was a lot
Did
of
you,
them.
in the thesis, did you manage to actually apply some modeling to some of these other?
No, that was
there
like
not
the
enough.
original idea is that I would look at everything, but then I just didn't have enough time, and I also kind of wanted the story to be a little more specific. But I do discuss the eagles in particular in my thesis because there's some literature showing or suggesting that they're not predating the seabirds very often.
Okay.
So I talked about that a bit.
Do I have time for,
There's that one as well.
do I have time for,
Yeah.
Okay, last one?
Yeah.
You mentioned, and I thought that was one of the interesting, in the discussion, one of the interesting things about the potential specialization of individuals, and as we were talking, I was like, oh, can we identify individuals? And you said no. But how would you, if you let's say you're studying a PhD tomorrow, and one of the questions is, do we have individual preferences for puffins, or do you have some individuals specializing towards that? How would you construct your
How would I look at
experiment
that?
to look at that?
I think I would need a lot of money
Oh,
first.
that's good. Imagine it's not a problem. Like, forget the money.
Okay,
Yeah.
imagining it's not a problem. I did read some stuff on trying to like track individual otters, and it's quite difficult. They don't want to wear anything, kind of, and they can't wear a collar because of their head shape. So I think using that would be quite difficult. There might be something you could do with DNA analysis of their scat, and to just see like who was on the island using that. Also, there's some studies that like tag them in like a visible way, like on their ear or something. So maybe that's something that could show up on the camera. Yeah, so maybe a combination of that with also like the camera traps, because sometimes you can't see males. There was one otter that had like a scar or some things like that. Yeah,
Thank you very much.
thank you.
Yes, thank you for your nice talk and for the way you try to use a very clear visual support for the discussion. That's really nice. I was first. There is a first question on your results. You showed that the puffin beds were at very different frequentation of
Very
the,
different ones, sorry.
of the abundance of the
Yeah.
of the predators. They were very different from one puffin bed to the others.
Yeah.
I was wondering if you have explanation for that. Is that they are a pair on the cliff, or
Yeah.
it's
Yeah, the spatial activity that they're in some areas more often than
Yes.
others. Yeah. My idea with that is that some of the colony areas might just be located next to like a preferred movement route potentially, or it's located maybe next to like a freshwater source or something like that, because they are pretty consistently in a lot of the colonies there in the winter as well. And from like my understanding of being on some of these islands and my supervisor as well, they were like, there's not really much like for them in this area. So it is kind of weird that they're like there actually that often in the winter. So it might just be a movement route. Otters might be following each other through these areas, like especially during when they're trying to breed. That would be my idea of that.
And then on your hypothesis, you have this variation of
Yeah.
for each period.
Mhm.
It's the
It's kind
the
of hard.
scale is like a few days, right?
No. So, well, these are the detection frequency, like
Yeah.
the real
So
data.
that's the really
This is real. And I have month on the bottom just to sort of simplify it, but each like data point represents a day.
Yes.
Yeah. So it's by day.
But on your hypothesis, why you add some days
Here?
and then no? Yeah.
Uh,
In fact, it's not one day. It's like 2
Yeah,
days.
this is just like something I just went like this on.
Okay,
sort of.
so it has
Sorry.
no
I was like,
meaning.
this is fake. This is just higher, lower, higher was
So
sort of
it's-
what I
it's
wanted to
the
illustrate.
level that I should read,
Yeah.
not the
Yeah.
up and down.
Yes, sorry.
All right, that makes sense. Okay. And about the prey
Mhm.
for the otter, what is the usual
Yeah.
prey for the
So
otter?
fish is what they
Fish.
prefer. They eat eelpout and rockling. That's been shown a lot. But I don't have any studies from these specific areas on the fish that they're eating. So that's like what I've gathered from other studies
But
in UK.
they without preference marine fish as well as freshwater fish.
Yeah, so these otters can either be coastal or in freshwater.
Okay.
So these ones are coastal. So on these islands, there aren't large freshwater systems, so they're primarily foraging in the ocean.
Okay.
Yeah, but they can do either. Yeah.
Oh, thanks.
Thank you.
Okay, yeah, thank you. Otters are really interesting
Yeah.
animals. I like them a lot. So when you were out in the field, did you see them?
I saw one otter
Okay.
one time from really far away. This month I was up there again.
So,
Not near the
yeah.
puffins.
so, well, my question is, what do they know about the population of otters? Is it similar numbers in the different islands, or is it one island that is have more otters? And,
Yeah.
uh,
Yeah, I don't have the exact data
Yeah.
of the number of otters on these islands, but the idea is that we definitely know there's a few like breeding females and families on Yamsoya. And the other ones, we just know that otters are present. Like before we started the studies, like there's just otters there, but again, we don't know how many. And these islands are pretty small, so it wouldn't be a lot probably, but there also might be some that come, like the males might come over for a bit, and they can swim too, so.
and they don't know if the population is growing.
Um,
or the otter
no, I think-
population,
I think it would be- yeah.
Because then the pressure would be higher in the end.
Yeah, I think it would be limited by the size of the island. I'm not sure if it's growing though, or if it has the potential to grow further than what is already living.
Do you know how much they migrate? Because if you have otters inland, they can actually go
Yeah,
many kilometers.
yeah, there's
Even in
some,
the forest.
yeah,
Yeah.
there's some studies on that, and it sort of seems like there's certain otters that have like a territory they stick to, like especially the females, and they'll have like overlapping territories, and then the males will move between them, and some females might be more migratory ones, and some might be more stationary, so it's kind of like, yeah, it could be
But
anything.
it would also be interesting to know about these otters if they go to these islands more often.
Yeah, like
than
just to
in
visit.
other islands that doesn't have fossils.
Yeah. Yeah, I would be super interested to know that as well.
Okay.
Because they can swim quite far
Yeah.
as well. Yeah,
Thank you.
thank you.
I have a question too.
Of course.
Yeah, so you said, I think in your conclusions that you saw that this type of predation is probably colony specific.
Yes.
Do you think you can conclude that from the data that you got?
I feel like, at least from the ones I have, like for Runda, for example, like the pattern or no pattern is like extremely different from the other two. And then even between, like there were a lot of similarities between Hornoya and Yamsoya, but I think it is kind of specific in, like I think they're eating more on Yamsoya, it seems like, and there's also just so many other factors in these colonies, like the size of the island and like human disturbance and distance from land and like all of these things, and that's going to vary like between any puffin colony, I think. So I think it would be, I wouldn't want to like extrapolate this towards a widespread explanation that would fit everywhere.
Okay. And I say just one question, but just one more. Did you, by any chance, just out of curiosity, spotted? I mean, how many otters did you spot? Like,
So I
your
can't say
cameras?
like abundance, and I can't say how many otters. 'Cause I don't know if they're all the
Okay.
same.
Yeah.
But I had 1,590 detections
Okay.
after I standardized it for like trying to, like the inflation of seeing one like right after the other.
Okay. And
Yeah.
and how many detections you had of them actually consuming? seeds?
Consuming, I think it was, well, I showed the 11 adults and the 2 chicks. So those were the ones that I determined were like independent events, because sometimes you might see, like I saw one take a puffin and then 3 minutes later it goes by another camera, so I'm not counting that as 2, I'm counting that as 1. It could have been 2, it could have been a different otter, but just out of caution. That was
And
one.
you detect the otters using something else?
Just the seabirds, and then we had some cameras that I actually excluded. I ended up not using in my thesis that were in den areas and latrine sites, and in one of those they had huge fish,
Okay.
but that's all I saw.
But other species of seabirds as well.
Yeah, the guillemots,
And
the
the
puffins,
razorbills.
and the razorbills. Yeah, and then sometimes you could see it was eating, but I couldn't tell which one of those 3 things it was.
Okay. And were there gulls present in this?
Yes. There are gulls. Mostly on my Hanoi data set was where I saw gulls, but I also, like, I know that there are gulls on the other islands. I think that was just because these cameras on Hanoi were also closer to the gull colonies. And they take a lot of eggs but specifically gill, not eggs mostly.
Okay, thanks very much.
Thank you.
Can you go to the start those?
Hmm?
Go to the start. It's the
Yeah.
last slide. Last slide.
Okay, so I leave you guys with Paula Matilda. Are you ready?
Okay, hello everyone. So my name is Paula and we are moving to a little bit different topic now. So we're gonna discuss my thesis, which was about how structural complexity and spatial context shape predation-based ecosystem functioning across natural and restoration sites in the Dominican Republic. Okay, so let's dive in. So to understand the motivation why I did this research, we have to first look at the current state of our ocean. And like we all know, coral reefs are declining globally. And this is, when the corals are dying, this is causing also the structure, the loss of the 3D structure. This flattening doesn't only mean that we're losing habitat, but it also triggers the collapse of key ecological processes, for example, top-down control. To fight this, we have seen a massive rise in restoration efforts. But restoration often falls to something that we call the structure-only metric check. So this means that we track the success of the restoration, primarily measuring things like coral cover or the physical growth, and we expect that if we build the structure, also the key ecological processes will follow. But the science is showing us increasingly that this is not always the case, and the structural recovery can actually mask a functional recovery gap, meaning that the structure is there, but the energy transfer and the trophic interactions are not. So therefore, that all the defined ecosystem functioning, something that it's the movement and the energy transfer, the movement and storage of energy and materials in the reefs. So to measure this, to test if the reefs are recovering, we need to actually measure this movement. In this study, I chose to use predation intensity as a simplified proxy for this ecosystem functioning to measure this energy transfer. Moving on to the research questions, so I wanted to know to what extent variation in reef morphofunctional traits relates to predation intensity and predator richness across reef habitats. I also wanted to know which specific trait drives this variation, and if the distance to the natural reefs influence predation independently of the local habitat structure. Finally, we also explored if the restoration sites occupy similar structural and functional space to the natural reefs. So to test all this, we conducted this study in Bahia Ida in Dominican Republic, which is in the southern East Reef Marine Sanctuary. And in this map, you can see the study locations, which were Sombrero, here, and Reef Balls, which is here. And there were 6 sites representing a gradient in structural complexity. So the sites were natural reefs, which are with varying coral cover and structural complexity. And these sites were primarily dominated by Orbitella species. There were these restoration sites, so I want to highlight its restoration site, it's not restored sites. So these are new sites, and they have these Mars-assisted reef restoration system reef stars there, which are these structures that are developed by Mars Sustainable Solutions Company. And these are the steel structures that are coated with resin and coral sand. And in some sites, there is some little coral recruits in them, and also some fragments, but some of them are also empty, so there is no corals in all of them. I also have sand flats that serve as low complexity reference habitats. So how did I translate these habitats into data? So I was using structure from ocean photogrammetry, and I did scuba-based photography across the 3x3 meter plots you can see in these example models here. And I ensured the overlap of at least 70% of all the images. I then processed the images in Metashape to build scale-dense point clouds, meshes, and digital elevation models. After that, I use different R packages to extract the structural metrics from these models. So the variables I measured were rugosity, which represents the surface roughness, fractal dimension, that measures the complexity in different spatial scales, convexity, which describes the surface protrusions and depressions. I have height grains, which is kind of self-explanatory, but it tells you the minimum and maximum height, and shelter capacity, which was the difference between the mesh volume and the bounding box volume. So it tells you the empty space in the model. How did I then measure the predation? So I used something called squid pop assays, which is a standardized method from marine GEO. And simply, you can see what it is here. So it's just a 1.3 centimeter piece of dry squid that is tethered to a 60 centimeter fiberglass stake. And I had all these stakes, and I put 25 of them in all of my sites, arranged in parallel rows or within the reef structures, as you can see the deployment here, spaced 1 to 2 meters apart. After that, I recorded the presence of the bait after 1 hour to get the immediate predation, and after 24 hours to get the overnight feeding as well. From this, I could estimate the predation intensity. I also have GoPro capturing the first hour of the deployment, and from these videos, I was able to identify the species actually driving the bite removal, and from this, I was able to estimate the predator richness. So moving on, I had 3 sites across 3 habitat types representing a gradient in structural complexity. For a total of 18 squidbot deployments, and this was done in 3 sampling grounds in 3 weeks intervals. In addition, I did 3 replicate 3D models of each site, and from these replicate models, I was able to get the average score for each structural metrics per each site. I then analyzed my predation data in R using binomial GLMM, where I had my different structural complexity metrics as fixed effects and distance to the natural reef, which I derived from the coordinates. And I also accounted for the non-independence by including the site and sampling period as random effects. For the predator richness, I used Poisson GLM, and I used the visible squid box in the video as an offset term to account for the different visibility in my videos, because in some videos I was able to see much more squid box than in others because the visibility was changing. Okay, so now on to the results. So let's look at first the physical environment. So here you can see all my structural metrics across the different habitats. And as expected, the sand flats were getting low values in height, rugosity, and shelter capacity. These were my low complexity reference habitats. But we see something interesting here because for fractal dimension and convexity, the sand flats are actually getting high values of values comparable to reef stars and natural reefs. And this could be that maybe sand flats appear structurally very simple in larger scales, but some of my metrics were actually measuring fine scale irregularities. And since sand can also have little dunes and ripples and things like that, so that's why some of my metrics were getting values like this, which was not expected. However, for my research question, this is probably not really relevant since the fish cannot hide in the sand. But this could be important for some other organisms like benthic organisms. So here is the multivariate structural space, and you can see here that the PC1 was driven mainly by rugosity, height, and shelter capacity. And you can see that the sand flats, they're occupying their own distinct region. They are the green dots. And we're getting consistently lower structural complexity values, whereas you can see that the blue and the orange dots are kind of overlapping, which might indicate a successful recovery of some of their structural characteristics. As for the predation results, so shelter capacity proved to be the strongest structural predictor of the 1 hour predation. So it is possible that refuge volume may be more important than the surface roughness alone for this question in hand. And you can see in this figure here the modeled relationship between shelter capacity and the predation probability. So it's increasing with the shelter capacity. The 24-hour data, we could see that the predation was high in all my sites, so there was no significant differences between the sites anymore after 24 hours. The predation intensity and predator richness also decreased with increasing distance from the natural reef, as you can see in this orange graph, so they were decreasing. And finally, the predation richness, it ranged from 0 to 4 species, and it was heavily dominated by red band parrotfish. And I think you can see that there is like a small increase in the predator species per squid pop also with the shelter capacity, but most probably these results indicate that the predation was driven by just a few key dominant general species and not maybe so much high biodiversity, since the richness was low across all sites. Here is the canonical analysis of principal coordinates, and it's constrained by the habitat to kind of see and visualize the more functional space. This is overlaid with the predation after 1 hour, so the color of the points is showing the superiority. And we can see here that the structural similarity did not consistently translate into similar predation intensity, and there were some differences there. So, what does it mean? So, landscape connectivity likely matters. So, restoring structure alone may be not enough, and maybe when planning where to place the restoration structures, it's important to also ensure the trophic connectivity between the sites. As I was mentioning earlier, the sandwells were showing high fractal dimension but low predation. So why is this? Like I said earlier, the ripples in the sand likely do not hide fish, but this could be still meaningful for some other organisms. So it's important to design structures across multiple spatial scales, including microcrevices to large vertical relief, because different kind of metrics can be important for different organisms and different functional groups. Also, integrating process-based indicators is important to monitor whether the ecosystem functioning is actually genuinely recovering or whether a functional depth persists. So to conclude this, ecosystem functioning likely do not just arise from physical structure alone, but it is a complex interaction between the habitat architecture, the spatial context, and also the species-mediated processes. I want to say thank you, and I want to also extend a massive thank you to Fundemar for hosting and supporting the project, my supervisors, and also the IMBRC consortium for making this research possible. And now here you can see the red band parrotfish. Really happy with the squid. And yeah, I would like to now open the floor for questions.
Yeah, I can start. Thank you for the interesting. I was thinking about these structures compared, for example, with the sand, because when you put the food on the sand, they were also disappearing.
Yes.
In the set after 24 hours, there were no difference between sand and the structure.
So you can see here actually the
Yeah.
results. This I didn't show earlier, but
Yeah.
so this is the proportion of the squid pops.
Yeah.
And so you can see in the first hour there is a big difference. So the predation in the sand is very low,
Yeah.
and it's like high to moderate in the natural reef and restoration sites. But yeah, after 24 hours, the one of the sand sites was still like lower, but it kind of saturated. So this could be due to like nocturnal feeding, like because of course I only have the camera recording the first hour, so I don't know who was then eating. Was it the same species? Was it different species? And also maybe there is connectivity between the sand flats, so maybe the fish was
Because
in.
if you were not knowing anything, you could say that, well, what's the big thing with the structure? Because the sand is also having the fish.
Mm.
But on the other hand,
Yeah,
the short-term effect was
Yeah, and you
strong.
can
So
also-
maybe the fishes are they more into this structure than
Mm.
during the day, or
Yeah, and you could see also when I look at the video, so I looked how many hours of the video, so you can, at least in the first hour, I was able to see that it was like lots of less fish in the sand. So this was like, I could visually like confirm this. And you can see also that the species in the sand were different. They were like flatfish and things like that, whereas in the reef structures and in the reefs, I had like parrotfish and triggerfish, which I did not see in the sand flats. So it would be interesting to know who was eating them at the end, but unfortunately 24 hours of proper footage is impossible.
so. And this structure, they are for like instead of the reef,
Yeah.
or is they in the end going to be a reef again?
Yeah, so the idea is that the reef will, maybe you can see it there. So the idea is that they will be like planted with coral recruits, and
Yeah.
over time they will like
Okay.
grow over them. But right now the coral recruits there are like tiny little polyps, and it will take obviously years before it's in that stage. So this was more just to see like, because the idea of these structures it is to bring 3D structure into the
Yeah.
site. So this was.
But it's two functions. In the short term, they will act as a 3D structure,
Mhm.
but hopefully in the end they
Yeah.
be
Because
a
also
new-
for the larvae settlement, it's
Mhm.
important to have, like naturally the larvae like needs something to settle,
Yeah.
but also they are doing sexual reproduction in the labs where they use. I don't know if there's a better. You can see these little stars.
Yeah.
So these are for the settlement. So these are actually in the tanks in the lab, and the larvae settles in them. And once they are big enough, they are then attached to
Okay.
the reserve.
Thank you.
Yes, thank you for your presentation. It brings hope with this kind of reefs, reef stars that you presented. I wanted to ask you, how do you see the connectivity? Because you are discussing connectivity, but it's like if you, what do you consider connectivity? Is this the way that fish go from one side to the other? Is it what you?
I think like I was trying to be careful to use the connectivity and more like spatial context for the reason that what I used was a proxy for connectivity, of course, because I was just measuring from my coordinates like the distance. So this is to proxy like to maybe see like if the distance to the nearest natural reef is, I didn't measure directly connectivity, and I think this is important to distinguish here. But what I mean in this context is the tropic like connectivity, that maybe like if there is long distances, maybe the fishes are not traveling like between the sites. So it's possible that if there is a source reef nearby, maybe the fishes travel more easy also to the restoration site versus, I think in the map I have here.
Yeah.
So in this it looks like they are really close by, but yeah, they are close by, and these are like very close by, and the predation was also like, it was like higher there, and you could also, by visually when I was diving there, I could sense this, that there was like more fish, and also there was more like species there. Whereas the reef balls, the restoration site is here, and the natural reef is here. So it's a longer distance, and you can kind of see this. So it is possible. I don't know for sure, and I think this is something that needs further research. But it is possible that maybe if the distances are longer, maybe the fish are not like moving so much, and it's easier for them to move to the closer side. So I think by positioning their structures near to the reef, this could be useful thing. But of course, it needs to be researched more.
And with your two study sites,
Yeah.
You did you study the differences between, for instance, the natural reefs versus reef star, like pairs of samples if you pair them from the same sites, or did you try to check if it's very similar from one
The
side to
predation or the structure.
the structure, I would say, but maybe if it's predation, tell us everything.
Yeah, so like for the natural reefs, it was all like kind of mixed. So I did compare it, like if the structural metrics have difference, and they were very similar. For the reef star sites, there was differences, and this was because you can see in these models, this restoration site also had these reef ball structures that have been there a long time before, and we included them in the 3D models because we thought that this might also have an effect for the predation, because they might offer additional shelter. So that's why I included them. But of course, this was changing the results because this thing has a lot of space inside, so shelter capacity increased. Also, the height difference was different. So this was causing a difference for the restoration sites. As for the predation, here you can see, it was really similar in the natural reef. Like there was not like big differences. But again, this was the reef balls sites, which was further away from the source reef. So there we could see a lower predation. But it could be for other reasons. I don't want to state that it's because of that, but it could be at least like to contribute to the distance. But there could be also other reasons, and also like human activities. This other place was like really close to the shore. Also, like there can be other reasons why the community composition, for example, is different. So.
And on your graphs where you showed the results with the lines you have, yes, on those graphs, the thickness corresponds to a sort of significance of your results. What is it exactly?
So this is
you
the
have the trend, and then you have a plus and
Oh, this is the confidence section.
yes,
Confidence interval, 95% confidence
and that's
interval.
the 95% confidence, right? Okay. And can you tell us a bit about the predator model that you used?
You in
For the fish, the GLM.
Yeah.
How do you calculate the predator richness? Because I haven't gotten that.
the Poisson GLM.
Yeah.
I choose. Yeah, it was done in R, but I used to use the Poisson version because it's a count data. So I counted the species. So that's why I chose to use Poisson. And GLM was because I did try to include like random effects as well, but they did not improve the model. So I ended up only using a GLM, which was the strongest model. I had the structural complexity and the distance to the natural reef also in this model. And then I used the offset term for the visible squid pops because like some of my videos, like it was really bad visibility, so I could only maybe see 3 of the squid pops, and then in a nice day there was 15 of them. So of course the sampling effort is different, so I had to account for the sampling effort.
Okay. Thank you.
Thank you, Paula. I have a few questions. First of all, what is what is to you, like without referring back to the slides, what is habitat complexity?
What?
What is habitat complexity? And without looking at the results of your tree habitats, how do you rank them in terms of their complexity?
I think I should talk more about the structural complexity, not the. I think habitat complexity can be something else, but structural complexity, like in this study, like I chose these 5 metrics to represent it. It's something that is multidimensional, and we cannot measure it with one metric only, but instead we need to, like define it in the large scale, in the smaller scale. We need to look into different things like protrusions, depressions. We need to use the roughness in different scales. We need to look at the height differences, and I think this was one thing why I chose, like based on the literature also, these metrics, but also because I think they were representing like kind of many of these features together. But it's something that depends, like I think structural complexity is something that it really depends like what we're also looking at, what is the ecological question in hand. Like for example, for me, like I was looking at fish, so the complexity that I'm looking is very different to if I was looking like benthic, small organisms where other metrics would be relevant. So I think it cannot be defined like this is it. It's a larger question.
There are other ways of looking at it than you did, like for example, looking at what are defining the basic structural element of your habitat and like applying things like richness and Shannon to these things also as possible. But there's many ways as you are, and depend on the question at hand. And second part of my question, once you have defined that, what, like again, without looking at your data and considering your research question, can you rank your tree habitats in terms of complexity a priori?
Well, this is again like it depends. Like, what are you looking?
On your within the context of your research.
I don't think if I can rank them, but like if we would just look what is relevant, like that's a really hard question. Like to rank it. I don't know if I can rank it because I do think that it depends really what metric you're looking and like what is relevant, because it's not just like one metric you cannot say that this is more complex than another metric, because like you can see in for example in this slide, like yes, you could say like based on this my restoration sites are the most structurally complex if you just look at this, but then it's also like other things that like play a role in this in my opinion. So also like some of my natural reefs also had really complex structures that were not found in the restoration site. So
well, that's exactly what I wanted to get to, because looking at this, we should conclude, as you said, that it's just the metal structure basically that's there in the restoration, which is the more complex, but I don't think it makes a lot of sense ecologically. So the underlying question is, are these the proper measures of complexity that you should be using, or could there be better ways? Because your measures collectively are saying, well, the reef star sites are the most complex.
yes, but I think what I was kind of interested is like which specific trait drives fish like predation. So this was what was interesting, and the answer from my research was shelter capacity. And this may be because maybe the shelter capacity provides refuge for prey and like hiding places for the fish. And like what I would like to study next, like and what we will likely also do in the future, is to see the quality of the shelter capacity, because like I was looking, so like shelter capacity was defined by the bounding box volume and the mesh volume. So it gives me all the empty space in the model, right? But I think that between the sites there will be differences. So some sites, like the actual shelter, like whether there is like larger crevices or smaller holes, like this, the quality of the shelter capacity will likely have
I
effect.
think it's missing that,
Yeah.
because you have lots of empty space, but it's
Yeah.
not well protected.
Yeah. And
As you're
this is
saying.
exactly the next thing to research and what I would like to do next.
Okay. So yeah, I think revisiting these metrics in the context of the specifics of your tree habitats, it would be, as you say, interesting.
Yes.
You have your canonical analysis. you went very fast on it, and you did not. I'm guessing the constraint
By the
variable
habitat type.
is the predators. Or-
It's constrained by the habitat type.
Yeah, but the what you're explaining is the predation and what you measured with
Yes.
the squid pops, right?
Yes, yes. So
Okay.
it's the predation, yeah.
Okay, because that was not.
This was to visualize kind of the functional space where the sites are, and then to see the predation. So you can see that in the tab one, the reef walls actually sites, and this is the sites with the large structures I was showing earlier. So it actually is high here, so indicating that the structure is high, but actually the predation is lower than all of this. So this is the thing that the structure doesn't always translate into the predation rates, and like you were saying, it's just metal structures. Like there are other things in play.
And did you think that here you used all your 5 metrics, right?
Yes.
Did you try just looking at maybe a simpler model of using just the first PCA or something like that that integrates your 5 metrics together, or
I was running the models with the PCA, but my supervisor and I discussed them. We ended up like running the models with we had it with the PCA, and then we run with all separate ones. And the best model that we thought was the one with the shelter capacity.
Very nice. Thank you very much.
Thank you.
Thank you. This concludes our session now.
start with the introduction already.
Okay. Okay. Thanks to you all for being here. We have now another session here, and I will be the moderator, Nicholas Wilber from the University of Oviedo. From the University of Göteborg, we got Per here, and also from the University of Oviedo, Alfredo Garcia and Gonzalo Machado as members of the jury. So I think we can start, and the first student will be Hussein Mohammed, please. Very welcome. As you know, it's going to be 15 minutes of presentation. We'll let you know when you have around 1 minute left. But you will tell me that, right? When it's 14 minutes or something like that? No, I will do that next. And and there will be other 15 minutes for questions or something. Okay?
Thank you all. Good afternoon. Thank you all for being here. My name is Ibrahim Hussein, and I will be presenting my master thesis on counting the ages in varying demographic parameters from scale analysis in the anadromous two-age chat, Allosaphorus. Two-age chat is an anadromous fish, which means it lives in the sea and migrates to the rivers for spawning. This species has declined across Europe due to the anthropogenic activities such as pollution, overfishing, and most importantly, building wires or dams that block the migration routes of these migratory species. But luckily, it's now protected under the EU Habitat Directive and a Berne Convention. For example, in Belgium, this species disappeared in the 1950s, but it has returned after almost 100 years of absence. But the return is really true, and the spawning has been observed. But there is a real big question: do we really know the demographic parameters of these recovering species? To know more about that, of the demographic parameters of this recovering population, this study focused to investigate the key parameters of this recovering population, such as the age, the structure, and the composition of the age of this population, how well they grow, and at what age they start reproducing. And most importantly, spawning frequency, whether they do spawning every year or they do skip a year. For the methodology, a total of 346 individuals were sampled from the sea scallop spawning grounds, which is the Belgian part of the Scheldt history, across 5 consecutive years, starting from 2020 to 2024, using double fake net, which is a type of funnel-shaped trap that allows the fish to go through but cannot go easily. For the nets, it was emptied every 1 to 2 hours to limit the mortality of the fish, and then the fish were transferred into a tank. We fill it with continuously pumped Styrian water to keep the fish oxygenated and alive. After that, 5 to 8 scales were sampled from each individual, and then were transferred into plastic bags, and then labeled with a unique ID, and then transferred into the lab. After that, the scales were transferred into the lab, which is the Outlet's lab at Ilvo. Then we started preparing the scales. We started from cleaning the scales with distilled water, and then gently rubbing between the index finger and the thumb to clear any debris or tissues that are remaining on the scale. After that, we mounted the scales under the slide glass, and then it was investigated under the stereo microscope connected to a computer with a delta box inside software that allows us to take the photo of the scale. After taking scale preparation protocol followed one of the most widely used references for the scale analysis of Twitchat, which was a billionaire at 2001. This is what Twitchat scale look like under the stereo microscope. As you can see, it has two parts, the anterior field and the posterior field. And you can see all these structures like the spawning mark and the annual growth rings. And then the images were transferred into the smart dot, which is the software that we are using to analyze the age of the fish. And the age reading protocol, 3 independent readers involved the reading of the scale. And then each reading was assigned to a quality score that ranges from AQ1 to AQ3. AQ1 was assigned if the scale was clear and the annual growth rings on the scale were clearly visible, and AQ2 was assigned if the scale was clear and or the reader has some dots on some annual growth rings, and AQ3 was assigned if the scale was completely unreadable and it was later discarded and was not included in the final model 8 assignment. After that, we did assign the final model 8 using a simple decision rule that says if all the 3 of us have the same age reading, that age was accepted as a final age model for that individual. And then if 2 of the 3 of us has the same age reading, then that age was also accepted. And if we have 3 different readings, then we use the median age. After that, we did some precision assessment to check if our reading was precise. We did some measurements like average percent error, which measures how different readers have different readings, and the coefficient of variation, which measures the average variability of the readings relative to the mean age, and percent agreement also measures how often the age the reading matched the final model age. We also used back calculation. The idea to use back calculation was to know the size of the previous age, and to do that we tested 4 different models: Delphi, Vesely, and the scale proportional hypothesis, and the body proportional hypothesis. And then Delphi was the model that was selected. And we also fitted 3 different growth models: Van Bortelen V and Gompert's logarithm. The choice of the 3 different models were based on AIC criteria. After that, we did a basic cohort analysis. The fish were grouped by birth year and cohorts. After that, growth and maturity were compared across cohorts. Coming to the results, the overall age structure of the population ranged from 3 to 9 years old, with age 5 and 6 dominating the catch. There was only one exception in 2020. That was in that year, age 4 was the dominant, and the oldest species recorded in this study was age 8 and 9, and they appeared only in 2024. For the length and the sex, females were larger than males on average, and the oldest species recorded were completely, exclusively female. Length increased 15 millimeter on each year, and males were 43 millimeter shorter than females on average. The median catch length increased from 395 millimeter to 435 millimeters. For how reliable is the aging, the 3 readers had about 60% on exact agreement, and for the overall error, average percent error was 11.8%, and the CP was 16%. In this case, scale aging is reliable for younger fish, but the older ones need to be read with more caution because the rings become more crowded and to differentiate becomes very hard. So age at first maturity, males mature at age 3, females at 4, and 95% of the fish matured at age 3 or 4. One male had already spawned at the age of 2, which was the earliest recorded in this data set. For the growth, as you can see on the left side graph, it shows the 3 different models fitted in this study. And as you can see, growth is fast during the first 2 years, and it starts to slow down as the fish grows older. The model which was the most fitted in this data was Gomport because it has the lowest AIC value, and the females were on the other side. We tried to fit the same data on to separate male and female, and then to show the difference of the growth between male and females, and we have seen that females are larger from age 1 until age 9. For the annual spawning, about 98% of the spawning spawn every year. Only 1.3% skip spawning. So skip spawning is rare in this population. For the cohort dynamics, there are 8 cohorts in this data set. So the growth, we compared the growth trajectories of these cohorts, and they were broadly similar. There were no differences, like larger differences between the cohorts. Coming to the discussion, there are 4 results in this study that point one direction. For example, fast growth and the large size and the early maturity and spawning every year are the signs of recovering populations. And in these recovering populations, there is a fewer individuals, there is a less competition. As there is a less competition, individuals grow very fast, and then if they grow very fast, they become mature in early ages. And how can we compare the population of the shelt to the other nearby or European populations? In this population, they are large compared to the other populations. For example, age 1 in this population is 117 millimeter. Compared to the populations of the Safen, Lorraine, and Cebu, which range from 85 to 100 millimeter in age 1. This population has a higher maximum length, which is 457 millimeter, and then that compares to the closest one, which is Safana, 417 millimeter. For the maturation, they mature at earlier compared to the other European populations, and the females outgrow males that match with the findings of the Korean population. The most importantly, how well scale aging worked in this species. To be honest, scales are far less precise than the otoliths, but the most references we have to compare is otoliths-based studies, and it's not always fair to compare scales and otoliths because otoliths are easier to read in terms of age reading, and the rings are more clear in otoliths. And the specific thing is that the scales of 2H are considered one of the most difficult scales to read for age reading. And they share the same burden with all other shared species. But there is a question here. We don't have any, do we have another choice so that we can do more to investigate the population demographics of this species? But the answer is simple, we don't have any other choice. Because this is protected species and it's recovering, So we can, if we need to use another structure for age reading, we need to sacrifice the fish. So we cannot kill this species. So it's protected species. But I think we can do one more thing to enhance the age reading and the limit human bias is to train AI models. So that could be the little improvements we can do. To conclude, this is one of the first baselines of this recovering population, and their recovery history is real. And this kind of species, which is anadromous, they always return when the water quality improves. So monitoring and further studies are needed here, specifically for the improvement of age reading and investigating demographic parameters of this species. And thank you for your attention.
Perfect timing, so we have plenty time for questions. If you want to start, Bert? Yeah, thank you very much. It's really interesting. What I miss a little bit here is that you do have a lot of data that could have been taken a little bit further, I think. What about the cohorts? Can't you, with some assumption, actually calculate mortality rates?
For the cohort analysis in fisheries, they mostly use a bigger data sets, mostly basic data. And when you wanted, what we wanted to try for the cohort analysis, we wanted to try how many of cohorts are lost in each year. Because in cohorts that were born in 2014, are they still exist in 2015? So what we wanted was to track these cohorts.
Yes, but if you have these data, there's some, with some assumption, you could maybe do a life changing of this.
Yeah, of course.
But you didn't discuss about that.
We didn't discuss about microfilms, but
because I think you still have even more interesting conclusions that you can actually draw from this data that you have. For example, you can calculate population growth rate to see if the population actually is growing or going down.
Yeah, I think we have growth.
Or maybe I'm wrong because you do have some limitations. But,
the growth we have.
Yeah, but that's the individual growth. I was thinking about
by
the by the number, if the population is growing by number. So, but otherwise, it's really interesting. And what do you think about the population? Is it growing? Can they see that it's more fish coming in? It's easier to get them in the nets every year, or what? Do the people say that did the field work?
Yeah, I'm not aware of the sampling, but I was not part of the sampling.
No.
But what I can see from data set is it's increasing each year. From 2020, it was kind of, if I correctly remember, 37 individuals. In 2020 was the same, but it has increased in 2022, and in 2024 was the largest. So it's increasing.
Yeah, that will depend on how much you put into the fishing as well, but still maybe good indication. But it's good to see that it works with the scales, so you don't need to do those.
Yeah, because we cannot do the outlets or the tapa, because otherwise it needs to sacrifice the fish. And this scale analysis remains the only non-level technique that can be used to investigate this population, and it's really needed to investigate this population because it's recovering, and this species was absent almost 100 years. So this is a crucial baseline study.
Okay, thank you.
Thank you. Thank you very much for the presentation. I like it. The layout is very clean and the visual aids are good, very good. Mm, I have a few questions about this. First, congratulations because it's very interesting to work with non-destructive samples for studying the populations organization today in this moment of global change. I also like the best fit models you are using. That is also good to see. And I have just a couple of questions. One is more curiosity because I checked for a loss of galaxy status in the IUCN red list,
Yeah.
but it looks like just general unknown
Yeah, it's
or
also
least concern.
yeah, it's also protected under two Berne Convention and the one I mentioned, that EU Habitat Directive. So countries have to protect and then do other studies for conservation purposes.
So it is protected in all European countries
Yeah.
or only in several?
European countries, all I know, because it's under EU Habitat Directive.
Okay,
Yeah.
which is good. So would you, do you know if there are scale collections in all European countries or scale collections are limited to some institutes or some countries?
These species are recovering and it's coming back now. All I know, the recent data of this species is that there was a recent publication. There was not like our data. They only have 2024. It was in Black Sea, I think, Poland. They conducted also some scale analysis, but they have very few individuals, like 3 or 4 individuals. So there is a recent data. That's the only one we have it. But other scale analyses are very old, so like 1900s.
Because, for example, in Atlantic salmon, we have scale collections in many countries.
Yeah.
So we can make comparisons between the status of populations in different countries and also use the scales as a source for DNA analysis,
Yeah.
for example.
This species was captured not to sample the scales. It was based on actually telemetry project that is going on that like to track this species because they are recently coming back for spawning. So there is one institution called IMBO. They conduct telemetry. When they are just for telemetry, you need to dissect and then transplant to devices that are used for tracking. So when they capture, they do the insection and then they put the tracking devices and then they sample. And then the samples were then transferred to the ILVO laboratory for collaboration. So that was the main idea for this species, was just telemetry purposes, but the scale analysis was just like for another studies.
So if I understand well, the species is coming back
Yeah,
now.
yeah, it's recovering.
So why do you think it's happening this? Are the water quality?
Yeah, mostly
general?
this species come back when the water quality improves. So it's mainly on environmental improvements.
Okay. So it's not sensitive to the temperature increase because
I would say yes, because when they do spawning, the actual temperature they need mostly is that from 10 to 12 degrees Celsius for the spawning. So they actually do spawning from spring to early summer. So if the temperature exceeds that, so that means they cannot do spawning properly. So they should be sensitive to temperature.
Oh, thank you. And my last question. So if they are coming back in the rivers, do you know where they were? They had the refuge.
Sorry?
They should have a refuge.
Yeah.
They should be somewhere. In what rivers they were like whatever. Because if the populations are recovering, this means that they were somewhere.
They mainly live in the sea and then they try to migrate into the rivers for spawning. But we don't have the data of the telemetry and we cannot say they traveled from this to this. All we know is that it was sampled in Scheldt River, it's 2 years old. And then that's all we know. But for the telemetry purposes, they have another publication, it's another different project.
Okay, thank you very much.
Okay, so thanks for such a nice talk. So I really enjoyed. So this was everything was based on data in one river. Correct me if I'm wrong.
Yeah.
Yeah. So do you expect any effect of if you compare the pattern of a scale reading for determine the age in a different river with a different?
Yeah, for the discussion we compared other populations in Europe and also in Morocco. Although the Moroccan population is quite different, there's two things. This species is heterochorus. When they spawn, they can spawn multiple years. But the Moroccan population, they only spawn 1 year, one time in a lifetime. So I think this population, what we see from the results now, that when we compare to other populations, this population is like larger in terms of like growth.
Okay, and have you tried, because you have there's some data, some scale collection plus your collection. Have you tried to extract DNA out of those scales? It's feasible, do you know? And maybe not you, but there's someone trying to do that. Because I mean, when you're referring demographic, it could be nice to have some independent source of data that can tell you about some connectivity, about some effective population size, and those things that could be interesting.
But we haven't yet tried that, but we only concentrated on the scale analysis for age and the spawning.
Out of curiosity. The other thing is just, again, out of curiosity, the way that you determine, because that's one of your contribution, the analyzing scale, reading for determine the age and growth and so on. So, in which way you start counting or analyzing the scales? Year by year or just randomly?
No, year by year. And individual by individual again.
Okay, but the reason why I'm asking this is that do you think that might have any effect of learning how to solve that the error in the process of doing the scale counting on determine the age?
Yeah. Actually, when you see the protocol you were using,
You can use the mouse as well.
The
when
way
you see the protocol, we were three different traders, me and two experienced lab technicians. So when I read all the scales, and then they start reading. So we didn't know each other. The readings all were like anonymous. So
okay.
I did, and then they do.
With the whole data set?
Yeah, the same.
Okay, that's with
So,
the whole data
and
set.
then- And then we use the decision rule for final model data assignment. So
Okay.
that was the thing to double check.
Okay, and at some point you mentioned something that for age 7 and 8, which was a little bit more harder to establish that because you have more error there. But also when you see the model for growth, I mean, can you go to also the confidence interval is much wider, yeah, due to the fact that you have really few individuals there, no? So definitely, I mean, that's having an effect. Not only the error percent that you might have, but also, I mean, clearly what you can see between the three models that you have there, it's clearly the scarcity of data there that is having a huge effect on that.
And for scale analysis, it's difficult specifically for this species. And when they get all the growth rings become crowded, and distinguishing between them is difficult. So that's why we couldn't agree much on the older age. But when it's like age 3, 4, it's like quite agreeable.
Okay, and just there.
Yeah.
So last one. If you have the opportunity to continue with this, what would you do?
if I have the opportunity to, actually, we are just working on this to make published, but we will try to do like some references. Like when you look this, we have again, sorry. We have what we call transversal groups, so we can count them, and then we can make easier like for the other coming researchers that they can just count the transversal groups, and then they say like age 1 and ring 1 and ring 2 and ring 3 is between that number of transversal groups. So we were just developing that kind of reference. So that will help, and also if we train a model like AI models, that could help limiting the human bias.
Thanks a lot.
That's
Great
the
job.
two things I could add, but it would be okay. Glad to be to add outlets, or that would be much easier, but we cannot do it in this.
Thanks a lot.
Thank you very much. The next talk will be given by Diana Marcela Rosero, please.
Okay. Good afternoon, everyone. Thank you all for being here. I'm Diana Rosero, and I'm presenting my master thesis project that is focusing the identification in silico monoterpene indole alkaloid genes and their expression under the marine diatom Thesis rotula. My supervisor was the Dr. Valeria Di Dato from the Stazione Zoologica in Napoli. Monoterpene indole alkaloids are a diverse class of secondary metabolites that are like super studied in terrestrial plants because they have like a huge pharmacological value. For example, they have like bioactive compounds such as bilastine that here we can see the pathway. This is just one route, but if we talk about monoterpene indole alkaloids, it's like a huge brand. We have two upstream isoprenoid like precursors that lead here. It can go to different like isoprenoids, but here we're going to the arenoid pathway, and after here, this is like a key precursor to go to all the monoterpene indole alkaloid, but our focus here is bilastine, so we can go here. As you see, like it's highly branches, it's like completely like compartmentalized, and it has different like limitations. So this opens the question, or like the door to explore non-traditional organisms that might have biotechnological potential. Here is where diatoms, marine diatoms, become important. Marine diatoms are photosynthetic microalgae that contribute to primary production, and their growth follows exponential growth curve, followed for a stationary phase. As in any other organism, they need essential nutrients. However, in their absence, or when they have like limited amounts, this can stimulate the synthesis of secondary metabolites. Thesis rotula is a centric marine diatoms, and is our, let's say, excellent model, exploratory model, because grow fast, can be maintained in cultures, and can actively like live with like this fluctuation of nutrients. And there is really important because we have available transcriptomic and genomic resources that in fact has been used already for the discovery of like different secondary metabolites. And in this case, we have a monoterpene indole alkaloid connection with the discovery of a sequence that is the enzyme in charge of producing the secologanin that, as explained, is the key precursor for all the monoterpene indole alkaloid. However, we have a knowledge, like a gap knowledge, that we don't know whether Thesis rotula contains partial or complete biosynthetic payload for this monoterpenoid. So this leads us to our richer question, that is, does Thesis rotula contains putative genes associated with the monoterpene indole alkaloid biosynthetic pathway, and are these genes transcriptional response to different abiotic conditions? So we have two objectives: the access and the identification of our putative genes in our genome of the Thesis rotula, and to evaluate their expression in different abiotic stress conditions. For this, the first objective, first we have a reconstruction of all the characterizing signs from the plant, the particular characteristics process, and then we have a screen with our genome through a lot of blasts, followed for like conservative domain validation to confirm whether or not or like sequences can have like these particularities of the enzymes that we're looking for. Additionally, we use some phylogenetic analysis to resolve ambiguity in some cases. For the in silico expression analysis, we have 3 different data sets. Each of it represents different abiotic conditions. The first one is for the silica depletion. The second one follows a vegetative growth and also a resin cell induction that was made by a complete nitrogen deprivation. And the third one, apart from the nutrients, it also evaluates the phosphate enrichment alongside with different temperatures. All the expression analysis was made for the entire transcriptome. However, my focus was the monoterpene alkaloid. So all the results and all the visualization of the interpretation of the results are only focused for the MIA. For our results, for the first identification of our genes, just to recap, this is the the precursor pathway. We have in plants we have both like roots, like 2 different roots that leads to the same these are the precursor for different isoprenoids, but our focus is this one that goes to the second part. This is the plant-like, but we have here the representation of the scheme of the pathway within the Thalassosfera genome. We can see here the identification of each sequence with the scaffold and the structure of the gene. And this is just important to say that we recover it completely, all of the 2 roots, and we have the enzymes to go to geranyl diphosphate that will be after used for the irinode branch that goes to the precursor. This is the plant line, just for you to follow which part we are, and that gives us the secolagamine. This is again the reconstruction pathway within Thalassosfera rotula. We also recover the entire pathway of all the enzymes. However, in this case, we have to use the additional phylogenetic analysis to kind of confirm or know whether our enzymes can be the ones that we're looking. That's why we have the boxes just identified. And for the downstream analysis, this is the plan. We have the same of multiple annotation that we have. However, here, this only reflects similarity within the family of genes. Doesn't confirm multifunctionality, even though we cannot discard it. Also, in this part of the pathway, we have some enzymes that we couldn't recover, but it doesn't mean that they're absent. We are comparing a plant with a diatom, so we have to consider the evolution of distance. So maybe for them, this they don't exist, the plant needs them, but maybe the diatom doesn't. Or we can have different, for other biosynthetic pathways, we can have enzymes that can perform this type of reaction that are needed to the production of these alkaloids. For the in silico expression analysis, then we have here, this is a control condition, this is a heat map. We have here all the identification of the genes followed with the Thalassosfera rotula genome identification. Just to remember, these 2 parts will be the precursors pathway, irinide pathway, and downstream pathway. The columns will be each part of the treatment. Here is a vegetative growth, so we'll follow from the day 0, day 3, and day 7. Day 3 is the initial of the exponential phase, so here they are growing. And the day 7 is the initial of the stationary phase, that in this case, the cells are more stressed. Here we can see that the pathway, when we see red colors, we can determine that we have positive values of expression, and where it's blue, we have negative values of expression. So we can say here that when this cell is growing and starting to grow in our exponential phase, this pathway is activated. So they're using it. We still don't know why. And when they started to the exponential phase, they also started to use it, but it's not that the magnitude is not that higher as when they start growing. If we compare it now within the abiotic stressor conditions, we have the silica depletion. In here, we couldn't find, we compare it to the control, we didn't have significant results. However, this condition is really important because it was the first analysis that I told you that we found different secondary metabolites, and the second organic, it was under silica depletion. And we can see here there is a positive trend in one of our precursors, that is the non-mevalonate pathway and the iridinoid pathway with the secologanic that is like really high expressed. Here we can see the phosphate enrichment along the temperature. We have control condition at 10, 25 degrees, and the phosphate enrichment at 10 and 25 degrees. Here is particular, you see that in control condition they prefer this route for the precursor pathway rather than this one, that is the non-mevalonate pathway. But the downstream pathway, we can see that it's not activated, but the pattern changes if we follow the phosphate. Here it could be like not activated, however the downstream is more activated. Maybe it's easy to visualize here in this interaction dot plot. We can see non-mevalonate pathway that is downregulated, but downstream pathways are upregulated. Here we see the resting cell induction that it was with nitrogen depletion, completely deprivation. And if you remember the control, we can see day 0, day 3, day 7, there is the growth, but then we reach the resting cell state after 1 month. We see that even in the day 3, they see like more or less the pathway is active. However, how we go down or like reaching like this status, they start to go down. You can see it here more clearly maybe when we have a complete downregulation of the entire pathway for both like in the day 3 when they start growing and the day 7 when there's more stress because of the stationary phase. We can see here a particularity of this enzyme that is upregulated, this gene. That is the cyclooganin synthase, that is the one of the cyclooganin product. This enzyme is a cytochrome 450, and in this particular pathway, they perform oxidation reaction. So maybe because they're stressed and they try to survive, they somehow use this enzyme in the survival. So, sum up. We have, for the putative genes, that we recover the pathway from the precursor pathway until the biblastin route. Here we can say we have more confidence with our results, but when we start down, we have started to have like a little ambiguity with some enzymes that could or could not be absent. And with the transcriptional responses, we have the silica, even though it's not have significant, if we compare using a control condition, we still have an active for the precursor and iridinoid pathway. When we have the resting cells, it's like completely downregulated. The cells are like trying to survive. They don't produce secondary metabolites, not at least in this route. But in the phosphate enrichment and with temperature, we have the strongest response, especially for the 25 and 10 degrees. We have to take into account that we have limitations. We are comparing a plant with a diatom. So we have this evolution at distances. So that's like one bias that we have of the references. We have functional ambiguity with like this large family that we can, until we don't do like enzymes like of the activity, we don't perform like it says about this, we cannot confirm like whether our enzymes can perform multiple multifunctionalities in different like reactions. And also we have everything is in silico, so we cannot establish like any metabolic flux, or we cannot say that we have like alkaloid production. So to conclude, these results provide the first explanatory assessment for all monoterpene indole alkaloid biosynthetic potential in the Thalassophila rotula diatom. The candidate evidence decrease as we go further in the pathway, and these putative genes are abiotic stress regulated in a condition-specific way, meaning that in this, depending on which condition they grow, their transcriptional response varies. It could be up or could be down. For future analysis, we have that we still have to make more cultivation on the target condition, prioritizing phosphate enrichment in the 10 and 25 degrees, follow for our target metabolomics and our alkaloid detection, so we can say like yes, our Thalassophila is producing it. follow for the enzyme-specific functional assays that will tell us if like actually these putative genes go further to enzymes and like are active acting, and why not comparative genomics as well if like needed. And that will be my master thesis. Thank you very much for being here and for listening. I wanted to thank my supervisor or my friends, my family that are here. And if you have any question, I will happy to answer.
Perfect timing, we have time for questions, so Gonzalo, you want to start?
Yeah, well, yeah. First, I would like to thank you for such a nice presentation. I really enjoyed it.
Thank
Your
you.
enthusiasm, your tone of voice, so you really keep our attention. So that's great. So well
Thank
done.
you.
I also think that it's quite it's a huge study that you did. I don't know which part, if you did the whole study or which part was mainly your duty within this.
Everything's in silico, and everything I did it myself. But like, yeah, since the recovery of the sign, like all the data mining, all the expression analysis, it
So
was
and you
like,
start from transcriptome data.
yes, this,
Okay,
yeah,
good.
these are already available, already published, but this one is in process of being published, and they're available in my
Oh,
lab.
yeah,
Mhm.
of course, that's great. So one of my questions is, have you thought about, since you have the transcriptomic data, have you thought, because typically when many different organisms, particularly eukaryotes, they are under really stressful condition. One of the fast response is, in some cases, it's just turn off or turn on or turn off the expression, but in some others, it's alternative splicing.
Yeah.
Because alternative splicing is much faster way to cope with changing the environment. So that's many people. So have you thought about, since you have the transcriptomic data, have you thought about see if there's some enrichment in transcript length, transcript of different length that could suggest an effect of?
Yeah, the main thing here is like, this is like no one believes that the atom can produce this type of secondary metabolites. Like they produce different types of secondary metabolites, but no monoterpene alkaloids that are full like for terrestrial plants. So the first idea is like, do they have it? Like might not even like, if we go into that, like we might not even have like a gene. So that at the beginning is just like, let's try to look what we can find, and after this we can like with the transcriptome data and everything, we can go into that. Yes.
I think it's more about, and also try to assess. I mean, I'm saying as a compliment. I'm not saying everything that you did. I think it's fully valid. It's great, but as a complement, as an additional line of evidence to see, okay, what's going on? Because even for those genes that might not be differentially expressed, you might have an enrichment of certain transcripts that might suggest alternative place in response. Also, when you have really stressful condition, also epigenomics play a huge role there to see how they are dealing with that. Yeah, so because, and as you know, also even epigenomic evaluation could be involved in alternative splicing also. So I'm saying just if you are trying to add up, so adding different line of evidence could be really, really great. And when you did the analysis, I mean, you did gene by gene. How did you treat in case that you have duplicate genes or gene families? How did you treat that?
Well, no, maybe here. Like, I'm gonna show you. I have like some extra. Like, what I did was like data mining. So I first did. I reconstruct the entire like pathway with characterized enzymes that are from the plant, and I made a local blast. Then I have like a long list of sequences that just homology based, but we cannot confirm just by homology. And I choose for the best hit, like evalues, the percentage of identity, But what it was like the main factor was the conservative domains here. So we have different sequences based because of the blast, and then we went to the conservative domain, and we see like I have even more sequences that belong to like shows what we're looking for example. But in some like some enzymes didn't like that. It's just they have like duplication maybe of like enzymes.
Okay, it's just
Like
how
a
you
sequence.
deal with that,
Yeah,
because
and also, like,
that
is it
might
like-
generate certain artifact in your
Yeah.
analysis. Because could be like if after duplication you have some sub specialization, and then what you might have is what you are assuming that it's the same gene that is expressed, but it's one of the copy of the other. So that's
Yeah,
why
and it could be
was
like
asking.
even fragments that can be like yeah.
Exactly,
Like this is like will be like further
Yeah,
to
yeah.
like go and like see like each one and see.
Okay. Yeah, once again, congratulations. I really
Thank
enjoyed
you.
it. Well, thank you.
Thank you. Well, congratulations for a very good presentation.
Thank you.
I also enjoyed how emphatic you presented the results, and I like the style for your speech that are, from my point of view, very good. For technicalities on the analysis, Gonzalo has already some questions. He asked you also about the part of the work that you did. So you did the in silico work, the bioinformatic analysis and all the investigation about the genes and the part of the genes that were transcribed. So I'd like to know a little bit more about the conditions of the experiments to understand what type of stress they are exposed to.
Okay.
Because I didn't get them
Yeah.
too
Like
much. Sorry, but
No, it's okay. It's just like in my thesis, it was only focused like the in silico, but I also work in the lab, so I also made like the culture, so I can explain it like better. You have the diatoms and you cultivate it in F2 medium, normal with all like the concentration that are like base of like the F2 medium. That would be the control condition. Then you play with the concentration of the nutrients. We know like I put it here. Oops. We know like we have like these that are like the essential nutrient for like the growth of the diatom, like silica, that is like for the formation of the phosphate. That is like completely important, and like we know about like nitrogen and phosphate. And what the idea is just cultivate it with a little bit lower amount of like the required like the normal of like silica, for example. Here was like 36 micromolar. The normal would be 50.
Okay, so they used less silica. than
Here
the
is
normal
less silica.
In
Yes,
that
in this
condition,
in silica
condition
depletion.
one.
Yes. This is the first analysis that was made. It was made in 2019, and that's where like everything of this story started, because they discovered like the secologani, and they were like, the secologani should be like, it's like the head of the monoterpene like a lot wife, like, or the atom can have that. So this is like really important. Here we have the control condition, the F2, and the resting cell induction was made with a completely nitrogen deprivation, so without nitrogen. And in this case, they cultivated with a phosphate enrichment. So more that were like a normal F2 medium would have.
This is a condition that they could be exposed to in polluted water, for example.
Yeah, like Napoli. At least like in front. They were, I think, they insulated, yes.
And the temperature at the same time?
See,
There
the
were
18
different-
degrees is like the control condition, like where they normally grow. And the idea was to evaluate like what is like in cold water, like more warm temperatures. So that's why there were like these two streams. And this one was analyzed only for the phosphate enrichment, so it was combined. So the culture, like you incubate like in a control environment. In the lab, they have standardized how they do it, like the lights, like all like the times. So they just put the chamber within these like conditions.
And what was the highest response? I mean, the what stress was the higher.
According to my results,
According to your results.
we have it here with the phosphate. Here. Phosphate associated with temperature. This is an interaction dot plot when we normalize the temperature that is like 18, like the references. So we're like taking this bias and comparing our temperature against the control condition at that time. So we can see in blue there is downregulated, and in red there is upregulated. And with the intensity of the color, it will be the magnitude of the expression. So we can see here like the log fold change that will tell us like this is like a easy way to see if they duplicate, if they quadruplicate the expression. So here we can say like with the size of the dots, like if they like duplicate, like if the expression like more. Here we have more intensity, so that's like yeah, they can like quadruplicate more, multiply. But here in the 25, we have more numbers of significant genes. Like if you compare it to the control, that means like they express more. Like their results are significant. So in 25 we have more dots, and here we have like more magnitude in this part of like the pathway.
So under warming, they will be like more stressed or respond using more genes, modifying the expression of more genes.
We can
That
say
makes
that.
sense.
We can say that it will be like, I don't know, say like prefer, but
Yeah.
it will be like, because phosphate, like they utilize it like for growing. So maybe they're happy. They're here like no stress as if we're like in silica depletion. They're like, oh, we need like they frustrate, like we need like to support. Now we have to defend. Here like let's duplicate. So it will be like a different like condition there, but like the happiness of the environment, even though there's stress because there's temperature differences. So maybe they have like potential to grow, but the stress will be the temperature is too hot. So maybe because it's too hot, it's like changing a little. But and as well where it's like too cold.
Okay, thank you very much.
Yeah, thank you very much for that presentation.
Thank you.
It's a little bit outside my expertise, this, but when you say the alkaloids production in plants,
Yes.
yeah, it's common, and and they have multiple functions. You said. One thing that you didn't mention is what kind of things could they protect?
In the atoms or
Yeah, well, in general.
well, they like the secondary metabolites. They use it like for more chemical defense.
Yes, from
A stressor like in the water, like
yeah,
all
like
the
a razor, maybe. So my point here
but
is
even
that,
like abiotic stressor.
yeah, but my point is, have you ever thought about putting a razor into the?
Well, now that we know that could have like a potential, we can play more with like different environments.
Because that would be a really stressful situation
Yeah.
for algae.
Yeah,
And
there's-
since this is often related to grazing, defense against grazing.
Yeah, that could be yes. Like now that we can have like we see the potential. Now we can play with like more, even like more abiotic condition as well.
Yeah.
Like yeah, for grazers. Yeah, I don't know how. Well, yeah, I think. Yeah.
Yeah, there are certain ways to do that. Thank you. Okay, thank you.
Thank you.
Okay, thank you very much, Diana.
you.
Congratulations. The next speaker will be Alexandra Pina.
Yes.
The floor is yours when you can.
Thank you.
Good afternoon, everyone. Thank you for being here. My name is Alessandra Pina, and today I'm presenting my master thesis on mapping small-scale fisheries in European and neighboring waters. This work was carried out within the framework of the Mapas SF project and was supervised by Marca Rufino at Istituto Portuguese do Mar Atmosfera in Lisbon and co-supervised by Anna Laura Tassetti at CNR in Ancona. So let me start explaining what a small-scale fishery is, also known by the acronym of SSF. So small-scale fisheries relies on small vessels, which under the EU definitions are small boats under 12 meters that don't tow their gear. And despite their fishing capacity could be low, there's a huge number of them. Worldwide, in fact, 90% of the fishing vessels are small scale, and in the European picture, the situation is the same because the vast majority of the vessels are small scale, and together they account for almost half of the fishing jobs. So these are not marginal numbers, as you can see. They support coastal community, they feed people, and in some cases also are part of the local identity. And here's the paradox, because despite their importance and their numbers, yet we still don't know where the small-scale fisheries actually fish. Why is that? The problem is structural, because if on one hand we have a large-scale vessel that were always legally required to carry tracking devices such as VMS or AIS, so we always knew where they were operating. On the other hand, small-scale vessels were never required to carry them, so there's no obligation, therefore no trace of them. And it was estimated that in 2023, only 12% of the EU small-scale fishery were tracked. And there's this new regulation that will extend the tracking obligation to all the vessels in Europe. But as of today, the most common vessels in Europe are also the one we know less. And this is not just a technical problem, because if we can say where they are, where they operate, how much they are, we can really estimate the fishing effort first, but also we can really include them in the marine spatial planning and conservation decisions. So they need to be put on a map, and some mapping has already been done, but always at a local scale, so area by area, country by country, and each in its own way. So there's still no single comparable picture of the European situation. And that's where my thesis tried to build. By answering my research question, which was to what extent can we actually map European small-scale fisheries by integrating different level of different type of sources, and we will level of uncertainty we can do that. To do this, I also split the main question in sub-questions, 3. First one about distribution and density, so how this split is spread along our waters and our densities. Second one about data quality, so how much we can rely on this map. So where are the data gaps and the data-rich area? And lastly, how much these fishing grounds overlap with marine protected areas and vulnerable marine ecosystems. Now, coming to the methods, the first problem I faced was how could I compare in one single picture 34 countries that were collecting their data in completely different ways. The solution I found was to start by building one common grid. Luckily, Europe, as you can see from this picture, already divided its waters into blocks, the management areas. The ICES system does it for the Atlantic and the Baltic Sea, while the GFCM system does it for the Mediterranean and Black Sea. So what I did was taking this management area and cross them with the national waters of each country. So that at the end I had a given amount of units, and each unit is the slice of the national water that falls in that given management area. So in this way, all the countries were described in the same way, and at the end I had a total of 117 units in 34 countries across the 4 basins. Once the frame was built, I took it with the best information I could find. My main sources were 4. First, spatial data, so management areas, water boundaries, coastline length, bathymetry, all those information that helped me construct the skeleton that I just described. Second, and most valuable information I had was the expert questionnaire, developed within the MapSSF project by my supervisor and her colleagues, and it was local knowledge of the fishing grounds from local experts, so very valuable information. Third, to gather information about vessel data, I mainly consulted the official fleet registers, and finally, to fill the gaps, to gather more information, I also consulted the National Fisheries reports and peer-reviewed studies. Now, coming to the core of the method, for each unit, I built its fishing ground from the most reliable source available and going to a weaker one if nothing better has existed. I had 5 levels of sources. First, extra special data. So in this case, the expert was giving me a ready-made map of the fishing ground in a shapefile or geopackage format. And what I did was reading this file directly into R as a polygon. Here we have the example of Sweden. Where in this case, so I had the map, I didn't construct anything, and I used the file as it was provided. Second level, literature mapping. So in this case, the map maybe it was directly from an expert, but it was already studied in a published in a publication. For example, here we have the Adriatic coast of Italy. And what I did was taking this image, overlaying it into Google Earth Pro using reference points, and tracing the boundaries by hand, exported it, and imported back into R as a polygon. Lastly, the last 3 levels, studies and constructions. What I did was, in this case, I didn't have a map, so I had to build the fishing grounds using 2 parameters. First one, how far the boat goes to fish. Second one, how deep it goes. And what I did was intersecting these 2 layers with the management area and the national waters of the given area or fishing ground. The only difference between these 2 levels, these 2 parameters, these 3 last levels, is where these 2 parameters came from. In this case, directly from the questionnaire. So here we have the example of Greece, of how I constructed this area of Greece. Second one, literature parameters. So when the questionnaire couldn't cover this information, I consulted the literature. For example, here we have the coast of Puglia. And finally, when nothing else better existed, I used a regulatory default, which is, to be honest here, the limit where trawling is banned. So it's not where small-scale fisheries actually was observed. It's more, they also can go beyond this limit sometimes. So that's why it's the weakest source of information I had. The key point is that at the end, for each polygon, for each unit, I built a polygon, and each polygon carries a confidence label from high to low, based on the source I used, as you can see from this thing. For the vessel counts, I basically did the same. I ranked 6 sources and used the best available one, from the questionnaire to official fleet registers and missing information if nothing better existed. And again, here also a confidence label was given to each source I used. And finally, I calculated an overall confidence for each unit based on the combination of the confidence of the polygon and the vessel confidence. And I took the minimum of 2. Why? It's a conservative choice, I know, but it's also the most honest one, because a perfect map of a fishing ground without knowing how many vessels there were is not that informative if you think about it. So now coming to the results, the construction gave me 106 out of 117 mapped units across Europe. For 10 units, that sits mainly in the Eastern Mediterranean here. I was unable to construct the polygon. For 1 unit, which was Belgium, it was confirmed that there's no small-scale fishery activity in that area, directly from the expert. From this first map, we can have a glance not only of where are the fishing grounds, but also how confident we are about this construction, following the color scale explained here. So dark green and light green with higher confidence, yellow, orange, and red with lower confidence. For the units I could count, I mapped 59,268 vessels all around my study area. And their distribution is anything but even. In fact, I don't know if you can see the numbers in this map, but this map shows the number of vessels per management area. We can see with the darkest colors that the Mediterranean holds 70% of the fleet in just a quarter of the mapped area. In fact, the countries with the highest number of vessels are Turkey, Greece, Italy, and Spain, all in the Mediterranean, while the Atlantic and the Baltic together account for almost 30% together of the fleet. So we can say that the Mediterranean concentrate both the boat but also the management challenge, because 1/3 of the fleet in the Mediterranean is from non-European countries, so also where the data obligation are different and also weakest. Turning to the density, it was calculated by dividing the number of vessels by the coastline length. And from this graph here, we can see that in the x-axis, there's the coastal length, while on the y-axis, there's the density. And the bubble size are the size of the fleet for each country. And what stands out, it's a north-south gradient. In fact, the countries of the Mediterranean and Black Sea in orange and pink cluster higher compared to the Atlantic and Baltic ones. And also the contrast is pretty strong because we go from a 4 vessel per kilometer of coastline in Malta to a 0.04 in Sweden. And this likely reflects a different coastal dependency, but also different fishery traditions. And how reliable is this map? So as I told you before, I calculated an overall confidence by combining the two components and taking the weakest of the two. And even if with the strict rule, at the end, 68% of the units resulted with a high or medium-high confidence. So it's still a pretty solid, a pretty robust picture. And also the units that had a lower confidence are not random. They mainly sit in non-European countries, in the Eastern Mediterranean mainly, but also in the Black Sea and in the UK outside of Scotland. And so these gaps are structural because all of these countries are outside the data collection framework. Finally, for the calculation of the overlap with protected areas, at the end, 68% of the units overlapped with the marine protected areas, while only 5% of the units overlap with vulnerable marine ecosystems. And this makes ecological sense in one way, because most fisheries work in shallow waters where marine protected areas are, while vulnerable marine ecosystems are more deep sea. But this last result should be read with more caution, because my proxy-based approach doesn't really take into account, doesn't really represent deep sea small-scale fisheries, which is present in Europe, such in Spain and in the Azores. So this the last result should be read with caution in this sense, because it's partly ecological, but also methodological. So what this work delivers? For the first time, small-scale fishing grounds across 34 countries in Europe were mapped with a transparent method, delivering the first spatial baseline of with explicit confidence level attached to each estimate. And so uncertainty is leveled, is there, and of course, this doesn't replace the future tracking data, but it's a strong foundation for future spatial planning, and also, hopefully, if published, it's going to be also an updatable. It can be an updatable data set ready to be tested against 2029 tracking data. Okay. And thank you all for the attention. Okay, I would like also to thank my supervisors and all the people that shared their knowledge through the questionnaire. They were really helpful. And all of you people to being here. And my family. Thank you.
Thank you very much, Alessandra. 14 minutes sharp. Perfect. So now, Eva, if you can start.
Yes. So, first, thank you very much, because you made this difficult topic enjoyable, which is very good. It's difficult, and this is very good, because this topic is outside general the topic of fisheries and fishing management.
Yeah.
What I can see is that the data are very heterogeneous.
Yes.
So you have
Yeah.
a problem with
Yes.
data heterogeneity.
Yeah.
And here I see many contributors. Did you work personally with all of them?
I work mainly with my supervisor. That she had a direct. She was the one contacted direct the expert because during the I mentioned the map SSF project, and during the building of this project, it was also where the questionnaire was built. And so she was the one sending the questionnaire to all the people. So she was also the one that was contacting the people directly. But during my thesis work in Lisbon, I also analyzed the questionnaire of the answers. And sometimes I found that some people were answering to maybe the question of, are you able to draw the fishing grounds? And maybe they were answering yes, but then the data didn't arrive. Or so in that case, I also myself contacted directly those people, and finally, so in some cases, for Sweden, I remember Patrick Johnson was the one I contacted directly. Also for Bulgaria. So for some with some people, yes, I also was the one in contact with them. But
Yeah, because you have already answered my next question, which was what part of the data did you collect?
Mainly yes. So mainly the from the questionnaire, the answer that I was targeting were, do you have the fishing grounds? Do you know where they are? And second, also, if you don't know, are you able to give us parameters to draw the image? So mainly these. Also, vessel counts. Sometimes the experts were giving me also right information about the vessel count in a given area. So also that one was one of my targeting answers.
So the questionnaire was passed to these experts.
Yeah.
The experts.
Sent directly, yes, to 86 people were contacted related to, of course, the fishery area. And at the end, we had a total of, if I'm not wrong, more than 36. I think I have a map. Yes. Yes. So here we have the overview of the countries, the number of experts contacted per country, and also the spatial scale of the questionnaire response. Because sometimes the expert was an expert of a local fishery area. So, for example, in Spain, some experts were like an expert of the Galician or the Catalonia. Some of the questions were at the regional. Some of the answers were at the regional scale.
Because sometimes the gaps are due to particular organization in each country of the management units. For example, here I see a gap in the north between the Basque country and Galicia.
Yeah.
This is due to the type of organization.
Different from Iraq over here.
Because the competences belongs to the, in Spain we call it autonomies, so to the regions.
Yeah.
There is a missing.
In this case, it's just the expert, like they were able to give us very good information in the Mediterranean part of the Spain, for example. We had like I remember like the ready-made map from this area. So, but other like from this place and this area, it was more like literature consulting to fill the gap. where the expert couldn't reach.
I understand the difficulty of
Yeah.
all this.
Yeah, it's a lot of.
Yeah. So, congratulations for your work. Just a couple of suggestions
Mhm.
if you want to publish on this.
Yeah.
First, this can be published in open access databases. Like you don't need to publish a typical article.
Yeah, no, we are thinking about a data paper, not a scientific one.
Yeah.
Like, yeah, because the core, I think, of this work is the methodology, like the data set that I was able to build with the questionnaire. I think it's really valuable, and it's really the valuable part is the fact that it can be updated with also more data that hopefully are going to arrive in 2029. Like in that case, we're going to validate, hopefully, but in the meantime, it can be.
So it can be already accessible.
Yeah,
which is very important. So I encourage you to put it in a public.
Yes, it's enough. That's the main idea. Yes.
The European one, for example.
Yeah.
EU data or one like this. Okay. Thank you very much.
Thank you.
Yeah, sure. Yeah, so congratulations. I really enjoy it, even though it's beyond my topic. Yeah, so, but I really enjoy it. Also, your enthusiasm, the way that you presented, so great. Congratulations. Really,
Thank you.
great job. I think it's really necessary, this kind of study, even though I'm not an expert on this, but clearly to show, I mean, you clearly point out the necessity of having more data about certain region. I think it's quite nice the way that you did. It's a lot of effort, of course, not only from you, but from many people involved. So it's quite a really good initiative, and I think that I fully agree with Eva that the idea of having this open access and the idea is to grow in knowledge. So it's really necessary, this kind of initiative. So congratulations about that. So awesome. So just out of curiosity, do you have the opportunity to somehow have some internal control? Let's say you contact expert from the same region that somehow to validate if the information that they are providing somehow match, or which is the degree that the information match between experts of the same region?
From you mean the national?
Yeah, so you contact experts from different parts of all along Europe, and then you get that information about the areas. So somehow it's like every time that you're doing some sort of questionnaire or questionnaires, that you need to have some internal validation to say, the match is quite, the degree of match between two experts on the same region is quite good, or which is the level of confidence of the match of two experts from the same area or same country, similar region at least.
Yeah, sometimes it happens. I think if I in Galicia was like that.
Yeah.
It was like 3 different experts from Galicia, and in that case, I was able to make a comparison from their data, like all together, and they were matching. In that case, they were matching. In some other cases, it's more about the vessels count. The match weren't, the numbers weren't the same.
So because all the information, sorry that I interrupt, all the information that you're getting in terms of country, the vessels, come from the expert, or from which other?
Sorry.
Can you? So maybe I skip.
Yeah. So these were the main sources I had for the vessel, the gathering of the number of vessels. Questionnaire was the first one because it was local knowledge, so pretty strong. Then the FDI dataset, UFIT registers and GFCN register are all datasets that I was able to consult to see the number of vessels, and they're all they were all open data source, so open to everyone. And what I did was just the second most valuable one is the FDI one, because it was giving me direct estimation of the number of vessels per sub-area. So they were matching directly with my units.
Okay.
So in that case also I had
So just again out of curiosity, so is there any seasonality on your data? So the way, what I'm asking is so the fleet, I mean, of course, are moving and they're moving depending on the target. The target are so it's fishery.
Yep.
So how, because the area could be even though it's one area, could be differentially explored.
Yeah.
And how that you think might affect your data, or how
Well,
useful could be to have into account this seasonality?
yeah, it will. I didn't do this study personally, but I think that once I think they're gonna be affected from seasonality, but here we are also counting vessels, not really. Once we are gonna try to estimate the fishing effort, I think in that case we're gonna be able to see if there's any seasonality change.
Yeah.
So in that case, I wish that I was able to also to estimate the fishing effort, but in this case it's just a matter of for now counting where they are and how many there are. But once
Yeah, because I would
It's the
think it's
picture-
more related to how you can apply these to some fisheries, poor data fisheries. So
Yeah.
if you have some way to make it a little bit more automatic, not automatic, but I
Yeah,
know
yeah,
it's
yeah,
not
yeah.
automatic, but how this type of study could, if you have estimate of effort, how can inform or somehow enrich the poor data fisheries?
Yeah, I think it's also gonna. I think it's gonna be more clear also with the landings data.
Okay.
So once we also have the landings data, we also have the day at sea. So once we also integrate those data, the picture is gonna be more clear also for that for those information.
That's great. Thanks a lot. Great
Thank
job.
you.
Yeah, thanks for this presentation. I'm really, really impressed about all the work you must have put down into this and put together everything.
Yeah.
Was that difficult?
At the beginning, yes, but just because I wasn't really like my whole path was fishery related, so I knew the topic. But working directly with this data was a little bit not traumatizing, but it was difficult at the beginning to find my way in the work. But once the picture was clear and the data were like all coming together, then I really enjoyed doing this. Like it's been fantastic. Like, really great time.
Now you have this fantastic data. You said you would like to go into the fishing effort, I guess. Yes. Yeah, because that's
That's really nice.
the
I'm so
most
curious about the
interesting, right?
comparison
But
with the tracking
what's
data.
your guess now? Is this
I mean,
important? I would say
yeah.
I come from Sweden
Yeah.
with a very, very few like these vessels.
Yeah.
So my guess for Sweden is that it's very, very low fishing effort from this.
Yes. Like here we have a picture of
Yeah.
Sweden,
Yeah.
and
But do you think it's
I think
the fishing effort can be important in some areas?
Can be sorry.
Can the fishing effort be important, like compared to the
It's always
big
I think
commercial fisheries?
I think it's gonna be like valuable to know how much fishing effort there's in the area. Of course, it's different also traditions and in Sweden.
Yeah.
So probably the effort is gonna be like way lower compared to a Mediterranean area.
I guess
where-
in Greece.
In Greece, it's gonna be way higher the effort there. But yeah, I think the data are gonna still gonna be very interesting and more accurate than for sure.
Will you be able to continue this?
Yeah, I would love to.
Yeah, but you can do it or?
Already? I'm sorry. Like now?
Yeah, well, do you know what can you continue with the project in some way? Or?
Well, I didn't really think, like I thought about it, but we'll see what the future. Yeah, we'll see.
I have a question myself. I see that you used EMONet database.
Yes,
For what exactly?
it was the MODnet, it was the bathymetry. Where is it?
Yeah.
No, it was
There.
here.
Yes.
It was here, yes.
Bathymetry.
Bathymetry, yeah. So-
I think they have also a product for bottom type that is quite accurate.
Yeah.
Have you looked at that? Because
for the deep sea fishery.
For everything.
Yeah. I didn't know, but that would be really informative.
So you can
because,
see you have
yeah.
more data on some bottom types
Yeah.
than other with other that
yeah, that's also
have
something that I
different
want.
species. So
Yeah, that's a thing that I want to see if it can be implemented in this way also, like to see also